Hello, Namaskara Karnataka. Hegidira ellaru. Welcome all of you yet again to an amazing class of Vedantu Karnataka. So today we are going to do molecular basis of inheritance part 2. Yesterday we have done part 1. If you haven't checked that video out, very bad. You have to check that video out, okay? Because this is going to be mission MBBS. How to get you into MBBS with free NEET classes, okay? Ivaga sidal chemistry nu nadita irutte, biology also will happen, physics also will happen. So first we have started with biology, high weightage topic. So molecular basis of inheritance, ಇಲ್ಲಿ ನೋಡಬಹುದು 40 marks ಪಕ್ಕ. So 40 marks ಪಕ್ಕ ಅಂದ್ರೆ ಏನು 10 questions ಇಲ್ಲ ಅಂದ್ರೆ 9 questions ಈ topic ಇಂದ ಖಂಡಿತ. In the past years also students almost 11 questions have come from this chapter. So ee chapter na underestimate ಮಾಡಬೇಡಿ. This chapter I will teach you little very slow. Like yesterday I have done very small portion. Okay, very small portion, and that topics also I have explained you very slowly and making you understand everything. Okay, notes are also shared on the channel. Okay, if you go to the channel yesterday's video, adal description box alli nodabodu session PDF link anta. Adan click ಮಾಡಿ ನಿಮ್ಮ phone number enter ಮಾಡಿದ್ರೆ automatically notes ನಿಮಗೆ download ಆಗುತ್ತೆ. Okay, good evening Anita, good evening Chetan, Sunita. Awesome. Okay, awesome. Others also, come on, put a lot of energy. I can see some names. Okay, yes. Okay, yes, quickly smash the like button as well. Okay, attendance mark ಮಾಡಬೇಕು, ಅದು ತುಂಬಾ important. Start ಮಾಡೋಣ second part. Okay, ಇವತ್ತು part 2 ಮಾಡ್ತಾ ಇದೀವಿ. ಸೊ ಈ ಟಾಪಿಕ್ ಇಲ್ಲಿ ನಾನು ಪ್ಯಾಕೇಜಿಂಗ್ ಬಗ್ಗೆ ಮತ್ತೆ ಡಿಫ್ರೆಂಟ್ ಟೈಪ್ಸ್ ಆಫ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ಸ್ ಬಗ್ಗೆ ಮಾತಾಡ್ತೀನಿ. ಓಕೆ, ಡನ್, awesome. Okay, let's start. Okay, so molecular basis of inheritance. So yesterday students we have done the basic. Okay, we have done the basic. Today we are going to do from packaging of DNA to yesterday last ನಾವು ಟಾಪಿಕ್ ಯಾವ್ದು ಮಾಡಿರೋದು ಚಾರ್ಗಫ್ ರೂಲ್ ಅಂತ ಅಲ್ವಾ. ಇವತ್ತು ನಾವು ಏನು ಮಾಡ್ತಾ ಇದೀವಿ, ಹಾಯ್ ಮಾದೇಶ್. ಸೊ ಟುಡೇ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಡೂ ಫ್ರಮ್ ಪ್ಯಾಕೇಜಿಂಗ್ ಆಫ್ DNA. Okay, how is DNA getting packaged? Okay, packaging of DNA. That's the topic. Packaging of DNA. So packaging of DNA means what? Just imagine. Okay, just imagine I'm giving you a long thread. Okay, now let's say mic ನೋಡಿ ಎಷ್ಟು ತುಂಬಾ ಲಾಂಗ್ ಆಗಿದೆ ಅಲ್ವಾ. I'm telling you this entire mic you have to fit into a small circle. Okay, which is 10 to the power minus 6. Okay, so you have to fit the entire thing into a small circle. Okay, small circle, that is what? Your nucleus. Okay, that is your nucleus. Wait, then move ಆಗ್ತಾನೆ ಇದೆ. Okay, so to this small circle I am asking you to put approximately how much students? Approximately 2.2 meter long DNA. Okay, long DNA into this nucleus. Now imagine you will be like, ma'am, ಏನು ಹೇಳ್ತಾ ಇದ್ದೀರಾ ಮ್ಯಾಮ್, ಇಷ್ಟು ಲಾಂಗ್ ಥ್ರೆಡ್ನ ಹೆಂಗ್ ಮ್ಯಾಮ್ ಅಷ್ಟೊಂದು ಚಿಕ್ಕ ಸರ್ಕಲಲ್ಲಿ ಇನ್ಸರ್ಟ್ ಮಾಡೋದು. So you will be thinking, what is this lady telling? Okay, so just like how you thought what I am telling, like that other people also thought, other scientists, other botanists, zoologists thought, wait, it is not possible, no. So there should be someone who is actually doing the packaging of the DNA. That is when they came across students they came across something called as histones. They came across something called as histones. Histones are actually positively charged protein. Okay, positively charged proteins. Positively charged proteins. What are positively charged proteins? Why do we need positively charged proteins? Okay, we need positively charged proteins because our very own DNA is negatively charged. Our very own DNA is negatively charged. So e negative charge obviously opposite poles attract, right? Let's say I am negative. Okay, I have one more person here, they are positive, we both will get attracted, right? I am positive, this person also is positive, we both will reject each other alva. ಆ ಥರನೇ ಪಾಸಿಟಿವ್ ಮತ್ತು ನೆಗೆಟಿವ್ ದೇ ವಿಲ್ ಅಟ್ರಾಕ್ಟ್ ಈಚ್ ಅದರ್. ಸೊ ಈ ಹಿಸ್ಟಾನ್ ಪ್ರೋಟೀನ್ಸ್ ಇಲ್ವಾ, ಅದು ಪಾಸಿಟಿವ್ ಚಾರ್ಜ್ ಇತ್ತು. ಸೊ DNA was negatively charged. DNA is negatively charged, so it will be easy for them to wind around each other. Okay, high attitude girl, so much attitude you have, name and all you have kept. So this histones, okay, these histones are a set of proteins, okay, they are a set of proteins that are going to help in the winding of DNA. Now see students, it's very simple. This is a long thread, okay, this is a long thread, okay. Now I have to make sure this entire thread gets into this, okay, small circle. So what am I going to use? I am going to use a set of proteins which are called histones. And how many histones are going to be there? Listen here, NEET point of view question. Okay, NEET point of view question nimige idu. No ma'am, I am one power ma'am, chumma I kept a name like that, okay. So yes, here histones are going to be actually H2, okay, H2A, H2B, H3, H4 and H1. So these 5 histone proteins together is going to do the binding of or packaging of DNA. But ma'am, yeng ma'am ivaru arrange agirodu 5 proteins anta helta idira. How are they going to arrange? How is DNA going to go around them? Aa tara one confusion irutalva. So ಇಲ್ಲಿ ನೋಡಿ, see, this is your one protein. Okay, let's consider this as H2A. Okay, H2A, one color. H2B, one color. Okay, ಇಲ್ಲಿ ನೋಡಿ, H3 one color. Okay, H3 one color. adamele H4 one color. Alva? Correct? Yes. So 4 proteins ide alva? Agreed all of you? So ಇಲ್ಲಿ ನೋಡಿ, shade madana. So we know that, okay, these are the proteins, okay. Thumbs up in the chat if you are understanding. Bega. Okay, done. This is the set of proteins. Now you will be thinking, ma'am, only 4. No, students, each of the proteins, Ilva, each of the proteins are going to double. They are going to come as a pair. They're like, I don't want to be single here. I want to come as a pair. So we will be like, okay, you come as a pair. It's fine for us. Now what can we do? Okay. So they are going to come as a pair. So H2A, 2 of them will be there. Okay. H2, H3, 2 will be there. HB, 2 will be there. Then you have H4, 2 will be there. Now you will be thinking, ma'am, you told H1 also is there. Where is H1? H1 will come later in the movie. Now, right now, what is going to happen? This, students, count madi. 1, 2, 3, 4, 5, 6, 7, 8. So 8 means, can I use the word octa? Yes, ma'am, you use whatever words you want. So it is going to be octa, right? So this word is going to be, this is going to be called a histone octamer. What is it going to be called? A histone octamer. Okay, octamer. So now this histone octamers are going to help in the winding of the DNA. Clear to all of you? Okay. So ಇಲ್ಲಿ ನೋಡಿ, let's say DNA ಯಾವ color ಅಲ್ಲಿ ಮಾಡಬೇಕು, white ಅಲ್ಲೇ ಮಾಡ್ತೀನಿ students. So now you see DNA how it is going to wound around each other. So it is going to go like this. Okay. It's going to go over this like this. Okay. Now if I draw too much, it will become too confusing. So I'll just connect it from here. Okay. And like this. Now you see the DNA has wound around. Okay. Wound around the whole thing. So this is how the DNA is going to wind around. Now what is going to happen? What is going to happen? Students, today one surprise is coming on the channel. Okay. Someone is coming. Who is coming? You have to guess. Okay. Someone is going to come to the channel ivattu. Okay. ಇಲ್ಲಿ ನೋಡಿ, ಹಿಸ್ಟೋನ್ ಆಕ್ಟೋಮರ್ ಅಲ್ವಾ? ನೀವು ಇದು ಏನದು ಮಾಲೆ ಕಟ್ಟೋದು ನೋಡಿದ್ದೀರಾ? For temples and all, like for pujas and all. Have you seen how they tie the flower garland? Flower garland tie ಮಾಡೋ ಟೈಮಲ್ಲಿ they'll keep one flower, and they tie one knot. One flower, they tie a knot. Why? Ma'am, because the flower should not move. Okay, flower should not move, and flower should not, let's say what? They should not move that side, this side, and the garland should look like a whole thing. Anta, alva. So, atara, okay, atara, ಇಲ್ಲೇನಾಗುತ್ತೆ, ಈ ಹಿಸ್ಟೋನ್ ಆಕ್ಟೋಮರ್ಸ್, ಓಕೆ, ಅರೌಂಡ್ ದ ಹಿಸ್ಟೋನ್ ಆಕ್ಟೋಮರ್ಸ್ DNA ಬೌಂಡ್ ಆಗುತ್ತೆ. ಅದಾದಮೇಲೆ ದಿಸ್ ಹ್ಯಾಸ್ ಟು ಬಿ ಇನ್ ಪ್ಲೇಸ್ ಅಲ್ವಾ, ಅದು ಮೂವ್ ಆಗಬಾರ್ದು ಅಲ್ಲಿಂದ. ಸೊ ಅಲ್ಲಿ ಮೂವ್ ಆಗಬಾರ್ದಂತ we will keep one person to lock these people. Okay, we'll keep this one person to lock this people. Okay, Chetan and Sunita, please don't disturb the class. You are disturbing other students. Okay, you want to message your friends, you message, but do not disturb another person, right? Because they are also coming here, and NEET students have to crack NEET. It's a most serious thing. Okay, so let's not disturb the class and let's not reduce the quality of our classes, right? Yes. Now look here. ಇಲ್ಲಿ ನೋಡಿ, ಸೊ ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಇಲ್ಲಿ H1 ಪ್ರೋಟೀನ್, ಓಕೆ, ಈ ತರ H1 ಪ್ರೋಟೀನ್ ಇಲ್ಲಿ ಬಂದು ಲಿಂಕ್ ಆಗುತ್ತೆ. ಓಕೆ, ಸೊ ದಟ್ಸ್ ವೈ ಸ್ಟೂಡೆಂಟ್ಸ್ ಈ ಪ್ರೋಟೀನ್ ನಾವು ಲಿಂಕರ್ ಪ್ರೋಟೀನ್ ಅಂತ ಕರಿತೀವಿ. ಓಕೆ, ಲಿಂಕರ್ ಪ್ರೋಟೀನ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಹಿಯರ್ ಯು ಸಿ ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ H1 protein, H1 protein, which is also called as the linker protein. What is it called? A linker protein. Clear? H1 protein or the linker protein. Clear? Done? Awesome. Okay. So, packaging of DNA, 2.2 meter long DNA. 1 second, students. Okay, so here what is going to happen, students? Here we have packaging of DNA. So 2.2 meter long DNA. Now one chick nucleus which is around 10 to the power minus 6. 10 to the power minus 6. Okay, so ಆ ಥರ ಒಂದು ಸ್ಮಾಲ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಅಲ್ಲಿ ಇನ್ಸರ್ಟ್ ಮಾಡಬೇಕು. ಇನ್ಸರ್ಟ್ ಮಾಡೋಕ್ಕೆ ನಮ್ಮ ಮೇನ್ ಪ್ರಾಬ್ಲಮ್ ಏನಂದರೆ, ಇಷ್ಟು ಲಾಂಗ್ DNA ಅಲ್ವಾ, ಇದನ್ನ ಇನ್ಸರ್ಟ್ ಮಾಡಕ್ಕೆ ಡೆಫಿನೆಟ್ಲಿ ಇಟ್ಸ್ ಗೋನ್ ಟು ಬಿಕಮ್ ಟು ಡಿಫಿಕಲ್ಟ್. ಸೊ ಅದಕ್ಕೆ ನಾವು ಎಕ್ಸ್ಟರ್ನಲ್ ಆಗಿ ಏನು ಮಾಡ್ತೀವಿ, ವಿ ಯೂಸ್ ಅನ್ ಹಿಸ್ಟಾನ್ ಪ್ರೋಟೀನ್ಸ್. ಲೆಟ್ಸ್ ಸೇ ನನಗೆ ಏನು ಮನೆ ಚೇಂಜ್ ಮಾಡಬೇಕು, ಓಕೆ, ಐ ಆಮ್ ನಾಟ್ ಏಬಲ್ ಟು ಡೂ ಇಟ್ ಬೈ ಮೈ ಸೆಲ್ಫ್, ಸೊ ಐ ಆಮ್ ಗೋನ್ ಟು ಕಾಲ್ ಪ್ಯಾಕರ್ಸ್ ಆ್ಯಂಡ್ ಮೂವರ್ಸ್. ಅವ್ರು ಬಂದು ಎಲ್ಲ ಶಿಫ್ಟ್ ಮಾಡಿಕೊಡ್ತಾರೆ. ಆ ಥರನೇ ಇಲ್ಲಿ ನಾವೊಂದು ಅಡಿಷ್ನಲ್ ಹೆಲ್ಪ್ ಯೂಸ್ ಮಾಡಿ ಈ DNA ವಿ ಆರ್ ವೈಂಡಿಂಗ್ ಅರೌಂಡ್ ಈಚ್ ಅದರ್. ಓಕೆ, ಸೊ ವಾಟ್ ಇಸ್ ಗೋನ್ ಟು ಹ್ಯಾಪನ್ ಸ್ಟೂಡೆಂಟ್ಸ್? Histone proteins, which are set of proteins, they are going to be positively charged and DNA is negatively charged. So negatively charged and positively charged proteins are going to, okay, yeah, thank you so much. So positively charged and negatively charged is going to wound around each other and like that. Now what is the set of proteins? This is only for NEET students. Okay, PUC students ಗೆ ಇಷ್ಟು ಡೀಟೇಲ್ ಆಗಿಲ್ಲ. This is NEET level only. Okay, H2A, H2B, H3, H4, H1. This is the set of proteins. So they are going to make what? They are going to make the octamer. So H2A, okay, H2B, H3, H4. Okay, H2A, H2B, H3, H4. So H2A ಎರಡು ಇರುತ್ತೆ, H2B ಎರಡು ಇರುತ್ತೆ, H3 ಎರಡು ಇರುತ್ತೆ, H4 ಎರಡು ಇರುತ್ತೆ. ಟೋಟಲಿ ಅವರೊಂದು ಆಕ್ಟೋಮರ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ. 1 2 3 4 5 6 7 8 8 ಅಂದರೆ ಆಕ್ಟೋಮರ್. ಓಕೆ, ಹಿಸ್ಟಾನ್ ಆಕ್ಟೋಮರ್. ಈ ಹಿಸ್ಟಾನ್ ಆಕ್ಟೋಮರ್, ಟು ಕೀಪ್ ಇಟ್ ಇನ್ ಪ್ಲೇಸ್, ಓಕೆ, ಟು ಕೀಪ್ ದ ಎಂಟೈರ್ ವೈಂಡಿಂಗ್ ಇನ್ ಪ್ಲೇಸ್, ವಿ ವಿಲ್ ಹ್ಯಾವ್ ಅನ್ H1 protein which is called the linker protein. ಕ್ಲಿಯರ್? ಅರ್ಥ ಆಯ್ತಾ ಎಲ್ಲರಿಗೂ? Students, you people are not sharing the video with your friends. Huh? You're not sharing your video with your friends at all. What are you people doing? What are you people doing? Tell me. Yes, share the video with all your friends and ask them to also join. Basic. One second. ರಿಫ್ಲೆಕ್ಷನ್ ತುಂಬ ಆಗ್ತಾ ಇದೆಯಾ? ಓಕೆ, ಓಕೆ, ರಿಫ್ಲೆಕ್ಟ್ ಆಗ್ತಾಯಿತ್ತು. ಓಕೆ, ಫೈನ್. Yes. Done, done, done. Share the video with all your friends as well. Okay. Now, done. This is done. Okay. Next node, na. So, this packaging of DNA is understood, no? So, what is going to happen, students? You'll know. So, there will be a long strand of DNA and then there will be... Now, I did not tell you what is a nucleosome. So, I am going to tell you that. Let's consider the entire histone octamer, ilva. We'll consider it as a single unit. Okay, we'll consider it as a single unit. Ee tara on single unit consider madi. Okay, fill madi le. Okay, done. Now, this fellow, again, repetition, repetition. Like this, many nucleosomes will be there. So, I am drawing it like this, okay. Like this. Many nucleosomes will be there, okay. Many nucleosomes will be there. Like this, the DNA's winding is going to happen, okay. So, this histone octamer, okay, this histone octamer, when it is wound like this, entire DNA winding is going to happen like this. Just understand that histone octamer plus DNA, okay, histone octamer plus DNA is going to be called a nucleosome. DNA plus histone octamer is called as what? It is called as nucleosome. It's called as what, students? It's called as a nucleosome. Artha aita? Done? Okay? Yes? Reflection barta idea do, chair do. Okay, done. So, histone octamer plus, so students, one nucleosome, remember this, NEET point of view very important, one nucleosome will have 200 base pairs. Okay, what will they have? One nucleosome will have 200 base pairs. Okay, NEET point of view very important. NEET point of view very important. Okay, done, understood. Now, okay, now, il nodi, DNA plus histone octamer is called as nucleosome. One nucleosome has 200 base pairs. Ivaga, now ee nucleosome, illandre histone octamer plus DNA combination, microscope, okay, field of microscope, andre, itara microscope al nodre, nimage ಏನು ಅರ್ಥ ಆಗುತ್ತೆ ಅಲ್ಲಿಂದ? You will notice that the structure, okay, the structure looks like a bead on a string structure. What does it look like? It looks like a bead on the string structure. Itara. Okay, itara. Illi histone octamers irutte. Illu histone octamer irutte. Illu irutte. Okay, like this. So, it looks like bead on a string structure under the microscope. Okay, very good. Okay, very good. Okay. Beads on string structure. Okay, beads on a string structure. Good job, good job. Understood? Awesome, awesome. Now, beads on a string structure is understood, nucleosome is understood. Now, students, sometimes, namage let's say additional help ಬೇಕಾಗುತ್ತೆ. ಇವಾಗ I am telling you, all of you, ನೀವು ಈ ವಿಡಿಯೋ ಪ್ಲೀಸ್ ಶೇರ್ ಮಾಡಿ ಫ್ರೆಂಡ್ಸ್ ಜೊತೆ, ಬಿಕಾಸ್ ಇದೊಂದು ಚಿಕ್ಕ ಚಾನಲ್ ಅಲ್ವಾ. ಸೊ ಈ ಚಾನಲ್ ಇಂದ ಒಂದು ವಿಡಿಯೋ ಮಾಡಿದ್ರೆ ಕೂಡ ಅದು ರೀಚ್ ಆಗೋದು ತುಂಬಾ ಕಷ್ಟ. Like everyone will not be able to see. So when you are like, let's say you are going to your, you know, offline coaching, illandre online elladru hogta idre, please share the video with your friends. Tell them like this one channel is there, nanagella artha aagta ide, neenu try maadu, ninage enadru doubts idre try madanta. Atara you can tell, okay. So ille enagutte nodi, this one nucleosome 200 base pairs is understood. Now students, namage atara one additional help bekagutte alva. So that time we are going to use NHC, okay, that is non-histone, okay, they are not histone, they are not part of the histone proteins, we already understood. Non-histone chromosomal proteins, non-histone chromosomal proteins. This non-histone chromosomal proteins are going to help us for that additional help, okay, they want to help us for that additional help. So non-histone chromosomal proteins help in packaging of chromosomes, okay, in higher organisms, okay, in higher organisms. 1 minute, I'll just remove this. Oh. Okay, now it's better. Now I'll just one second. Okay, look here, non-histone chromosomal proteins help in packaging of chromosomes, okay, in higher organisms. In higher organisms, okay, done, done? Clear? Yes, non-histone chromosomal proteins, okay, non-histone chromosomal proteins, they help in packaging of chromosomes in higher organisms. Done. Okay, now this is clear, almost we have covered. Now we have to study about euchromatin and heterochromatin, okay. Idu delete madbirtini. Euchromatin and heterochromatin. Now students, nodi, on chromosome alli, okay, in one chromosome, there will be regions, okay, just think like this, this is one chromosome, right. So, in a chromosome, there can be regions, okay, there can be regions where there is densely packed and where it is lightly packed, okay. I'll show you in a diagram, you will understand that. So, idu densely packed, alva, id nodi loosely packed, okay, again loosely packed, densely packed. So, in a chromosome, there can be chromatins, okay, which are densely packed or they can be loosely packed. Now, here if you see, in nodidre, ma'am, here this region is densely packed. That region is called as heterochromatin. That region is called as what? Heterochromatin. Heterochromatin, okay, is going to be obviously students densely packed, right? It is going to be what? It is going to be densely packed. Okay, now neeve heli on room alli, okay, on room alli, let's say only 20 people can fit in, okay, let's say ee studio dalli 20 people can fit in, but at that time alli 20 students ige instead of 20 students, let's say 50 students came. ಇವಾಗ ಕಂಜೆಸ್ಟೆಡ್ ಆಗುತ್ತಲ್ವಾ? ಎಸ್ ಮ್ಯಾಮ್, ತುಂಬ ಕಂಜೆಸ್ಟೆಡ್ ಆಗುತ್ತೆ ಮ್ಯಾಮ್. 20 and 50 ಸ್ಟೂಡೆಂಟ್ಸ್ಗೆ. ಇವಾಗ ಲೆಟ್ಸ್ ಸೇ ಸಡನ್ಲಿ ನಾನೊಂದು ಕಲರ್ಸ್, ಹೋಲಿ ಕಲರ್ಸ್ ಇಲ್ಲಾಂದರೆ ಏನಾದರೂ ಒಂದು ಕಲರ್ಸ್ ನಾನು ಡಿಸ್ಪರ್ಸ್, ಇಲ್ಲಾಂದರೆ ಈ ಥರ ಜಸ್ಟ್ ಐ ಆಮ್ ಜಸ್ಟ್ ಸ್ಪ್ರಿಂಗ್ಲಿಂಗ್ ಇಟ್ ಆನ್ ಪೀಪಲ್. Everyone are going to get colored, everyone are going to be stained. Why? Because they are tightly packed. ಅಲ್ವಾ? ಒಂದು ರೂಮಲ್ಲಿ 20 ಸ್ಟೂಡೆಂಟ್ಸ್ ಇರ್ಬೋದಾಯಿತು, ಬಟ್ ಅಲ್ಲಿ 50 ಸ್ಟೂಡೆಂಟ್ಸ್ ಇವಾಗ ಇದ್ದಾರೆ. ಸೊ ಡೆಫಿನೆಟ್ಲಿ ಅವು ಟೈಟ್ಲಿ ಪ್ಯಾಕ್ಡ್ ಆಗಿರುತ್ತೆ. ಸೊ ಟೈಟ್ಲಿ ಪ್ಯಾಕ್ಡ್ ಆಗಿದ್ರೆ ಸ್ಟೇನ್ ಆಗೋದು ಈಸಿ. ಓಕೆ, ಸೊ ಆ ಥರನೇ ಡೆನ್ಸ್ಲಿ ಪ್ಯಾಕ್ಡ್ ಆ್ಯಂಡ್ ಡಾರ್ಕ್ಲಿ ಸ್ಟೇನ್ಡ್. Darkly stained. Now densely packed, darkly stained, but here these people are transcriptionally inactive. Transcriptionally inactive. Understood? Yes, understood all of you? Bega video, smash the like button. Come on Karnataka students. Okay? Yes. Smash the like button. Break the like button. Break the like button. Okay, we don't need like buttons. Come on. Just smash the like button. and mark your presence in the class. It's like attendance, okay? College allo attendance kottira, illu atarane attendance, instead of attendance, just smash the like button that ma'am I was present. Adamele class admele comment ಮಾಡಿ, so I know your names as well. Okay, atarane connection build agado. So transcriptionally inactive. Now here nodi students, illi these are loosely packed alva. These are called as euchromatin. Euchromatin. Okay, euchromatin but loosely packed. So ange bariri, loosely packed, lightly stained, okay? So loosely packed, lightly stained, ill inactive agidre ill enagutte, transcriptionally active, transcriptionally active. Okay, done? Yes or no? Is this done? Transcriptionally active. Awesome, awesome. Done, all of you. One minute, students. Done. ಇಷ್ಟು ಸೈಲೆಂಟ್ ಸ್ಟೂಡೆಂಟ್ಸ್ನ ನಾನು ನೋಡೇ ಇಲ್ಲ ಲೈಫಲ್ಲಿ. ಎಷ್ಟು ಸೈಲೆಂಟ್ ಆಗಿದ್ದೀರಾ ಪಾಪ. Okay. Very good, very good. Like I want all of you to be like Anita, attitude girl, ಸ್ವಲ್ಪ ಆಕ್ಟಿವ್ ಆಗಿರಬೇಕು ಕ್ಲಾಸಲ್ಲಿ. Okay? Okay, done. Done, ma'am. Very nice, very nice. Yes. Done? Awesome. Okay, continue ಮಾಡೋಣ. So heterochromatin ಬಂದ್ಬಿಟ್ಟು densely packed. Densely packed ಆಗಿದ್ರೆ darkly stain ಆಗುತ್ತೆ. So ಇಲ್ಲಿ heterochromatin, please remember HD. ನನ್ನ ಕ್ಲಾಸಲ್ಲಿ ನಿಮಗೆ ಮ್ಯಾಕ್ಸ್ ಟು ಮ್ಯಾಕ್ಸ್ ಟ್ರಿಕ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ಆನಿಮಲ್ ಕಿಂಗ್ಡಮ್ ಚಾಪ್ಟರ್ ಇರ್ಬೋದು, ಇಲ್ಲಾಂದರೆ ಮಾಲಿಕ್ಯುಲರ್ ಬೇಸಿಸ್ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಇರ್ಬೋದು, ಹ್ಯೂಮನ್ ಫಿಸಿಯಾಲಜಿ ಟಾಪಿಕ್ ಇರ್ಬೋದು, ನಾನು ಮ್ಯಾಕ್ಸಿಮಮ್ ನಿಮಗೆ ಟ್ರಿಕ್ಸ್ ಯೂಸ್ ಮಾಡಿ I'll try to explain the entire concept, okay? So ನಿಮಗೆ ಆ ಒಂದು ಟೆನ್ಷನ್ ಬೇಡ, ಅಯ್ಯೋ ಮ್ಯಾಮ್ ಹೆಂಗ್ ಮ್ಯಾಮ್ ಆನಿಮಲ್ ಕಿಂಗ್ಡಮ್ ಫುಲ್ ಚಾಪ್ಟರ್ ನೋಡು. ಐ ವಿಲ್ ಟೀಚ್ ಯು ವಿತ್ ಸೂಪರ್ ಟ್ರಿಕ್ಸ್. ನನ್ನ ಕ್ಲಾಸಸ್ ನಿಮಗೆ ಎಲ್ಲಾದರೂ ಹೋಗಿ ನೋಡ್ಬೋದು. ಮ್ಯಾಕ್ಸಿಮಮ್ ಟ್ರಿಕ್ಸೇ ಇರೋದು, ಬಿಕಾಸ್ ಟ್ರಿಕ್ಸ್ ವಿಲ್ ಬಿ ಸ್ಟಕ್ ಇನ್ ಯೋರ್ ಹೆಡ್. ಬಟ್ ಆಲ್ ದಿಸ್ ಎಕ್ಸ್ಪ್ಲನೇಷನ್ ಯು ವಿಲ್ ನಾಟ್ ರಿಮೆಂಬರ್ ಸ್ಟೂಡೆಂಟ್ಸ್. ಆ ಎಕ್ಸಾಮಲ್ಲಿ ಅಷ್ಟು ಕ್ವೆಶನ್ ಸಾಲ್ವ್ ಮಾಡ್ತೀರಾ, ಫುಲ್ ಪ್ಯಾನಿಕ್ ಮೋಡಲ್ಲಿ ಇರ್ತೀರಾ. ಆ ಟೈಮಲ್ಲಿ ನಿಮಗೆ ಗೋಪಿಕಾ ಮ್ಯಾಮ್ ಈ ಥರ ಆವತ್ತೇನೋ ಒಂದು ಜೋಕ್ ಹೇಳ್ತಾ ಇಲ್ಲ, ಅದೇ ನಿಮಗೆ ನಾಪ್ಕೆ ಇರೋದು. ವಾಟ್ ಎವರ್ ಐ ಟೀಚ್ ನೌ ಯು ವಿಲ್ ನಾಟ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್. ಓಕೆ, awesome. Yes, that's the spirit. Thank you so much, dear. Okay, so illi you remember like this, HD. Okay, HD you remember. Why HD andre heterochromatin, densely packed, darkly stained. Okay, how will you remember here? You remember E and L, that is euchromatin. So HD, if you remember, it is more than enough. HD TV, okay, HD computer idea, HD photo, HD picture, HD send me HD images on WhatsApp anta heltiralva. HD, H for heterochromatin, D for densely packed, D for darkly stained. Other the opposite ನೀವು euchromatinal ಬರಿಬೇಕು. Other direct opposite euchromatinal ಬರಿಬೇಕು. Okay, ಇಲ್ಲಿ ನೋಡಿ, we are going to start with experiments. Experiments ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್. ಓಕೆ, ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್. ಸ್ವಲ್ಪ ಬ್ರೈಟ್ನೆಸ್ ಕಮ್ಮಿ ಮಾಡಬೇಕಾ? ಓಕೆ, ಇಲ್ಲಿ ಮಾಡ್ತೀನಿ, 1 minute. ಓಕೆ, ಇವಾಗ ಇನ್ನೂ ಸ್ವಲ್ಪ ಕ್ಲಿಯರ್ ಆಗಿ ಕಾಣಿಸುತ್ತೆ. ಎಸ್, ಸೊ ಈವನ್ ದೋ ದ ಡಿಸ್ಕವರಿ ಆಫ್ ನ್ಯೂಕ್ಲಿಯನ್ ಬೈ ಮಿಶರ್ and the proposition of principles of inheritance by Mendel were almost at the same time, but that the DNA acts as a genetic material, okay? DNA acts as a genetic material took long to be discovered and proven. By 1926, remember, by 1926, the quest andre on thirst, illa andre on need. Ma'am, ನನಗೆ ಇವಾಗ ಅಂತರ ಒಂದು ಕ್ವೆಸ್ಟ್ ಇದೆ, ನನಗೆ ನೀಟ್ ಕ್ರ್ಯಾಕ್ ಮಾಡಬೇಕಂತ. That means that you have that aggression, you have that search, you want that, you know, that willingness that okay, we have to find this. ಆ ಥರ ಒಂದು ನೀಡ್ ಬಂತು ಆ ಟೈಮಲ್ಲಿ. So that the quest to determine the mechanism for genetic inheritance had reached the molecular level. Okay. Now, previous discoveries by Mendel, Sutton, Thomas and Morgan. So Morgan, Sutton, Boveri, adella nimage principles of inheritance al gottagutte. So Mendel, Sutton and Morgan and numerous other scientists had narrowed the search to chromosomes located in the nucleus of the most cells. But the question of what molecule was actually the genetic material was not answered. So our confusion ittu, DNA agirbodu genetic material. Ivaga naan helidini nimage introduction alle naan explain madidini that no students, DNA is the genetic material for most of the organism. But our information 1926 alli clear agidilla, 1869 alli clear agidilla. Because everyone were trying to find out, no, what is the genetic material, who is the genetic material, who is that one cell that holds all the information. So ಆ ಒಂದು ಕನ್ಫ್ಯೂಷನ್ ಬೇಸಸಲ್ಲಿ ಅವ್ರು ಈ ಥರ ಒಂದು ಸರ್ಚ್ ಸ್ಟಾರ್ಟ್ ಆಯಿತು. ಓಕೆ, ನಮ್ಗೆ ನೋಡಬೇಕು, ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ಸ್ ಮಾಡಬೇಕು, ಅದರಿಂದ ಚೆಕ್ ಮಾಡಬೇಕು ಹೂ ಇಸ್ ದ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ಅಂತ. First experiment that happened was Griffith's experiment, students. Okay, Griffith's experiment that happened in 1926. Dates are important. So in 1926, Griffith's experiment was based on mainly what his experiment was based on Griffith's experiment one minute. So Griffith's experiment in 1926, okay. So I'm just putting the heading here for all of you to understand, okay, yeah. So it happened in 1926, okay. Now what was the experiment based on? Okay, what was the experiment based on? So Griffith experiment, student, it was mainly based on Streptococcus pneumoniae. Streptococcus pneumoniae. Streptococcus pneumoniae was the one who causes what? Ma'am, ille ide ma'am, pneumonia. It causes pneumonia. So pneumonia, students, it is a disease, okay, which is caused mainly from an like young spread agutte by the help of droplets illandre aerosols. Let's say one person has pneumonia, out sneeze ಮಾಡ್ತಾರೆ, ಆ ಸ್ನೀಸಿಂಗ್ ಇಂದ ಎಲ್ಲಾದರೂ ಒಂದು ಲೆಟ್ಸ್ ಸೇ ಒಂದು ಆಬ್ಜೆಕ್ಟಲ್ಲಿ, ಓಕೆ, ಒಂದು ಆಬ್ಜೆಕ್ಟಲ್ಲಿ ಬಿದ್ದ ಆಮೇಲೆ ಲೆಟ್ಸ್ ಸೇ ವಿ ಟೇಕ್ ದ ಆಬ್ಜೆಕ್ಟ್. ಸೊ ಆವಾಗ ಆಟೋಮೆಟಿಕ್ಲಿ ನಮಗೂ ನಿಮೋನಿಯಾ ಬರುತ್ತೆ, ಇಲ್ಲಾಂದರೆ ಇನ್ಹೇಲಿಂಗ್ ಆಫ್ ದಿ ಏರಿಸೋಲ್ಸ್ ಆಲ್ಸೋ. ನಾವು ನಿಮೋನಿಯಾ ಇನ್ ವೆರಿ ಸಿವಿಯರ್ ಕೇಸಸ್ ಇಟ್ ಈಸ್ ಬೇಸಿಕ್ಲಿ ಫ್ಲೂಯಿಡ್ ಫಿಲ್ಡ್, ನಮ್ಮ ಆಲ್ವಿಯೋಲೈನ ಅಫೆಕ್ಟ್ ಮಾಡೋದು ಲಂಗ್ಸ್ನ, ಓಕೆ. So here what happens, Streptococcus pneumoniae is the one that causes pneumonia. So he was working on Streptococcus pneumoniae and he noticed that there were two colonies that were formed, okay. So this is, let's say, one Petri plate, this is one more Petri plate, okay, two Petri plates are there. He noticed that two different colonies are growing, okay, two different colonies are growing. Illon colony, Illon colony, okay, Illon colony. So idara ಎರಡ್ colonies, okay, continuous ಆಗಿ, yes, very good, ಎರಡ್ strains ಇತ್ತು. 1928, good job, good finding, okay, 1928. So Illon colony ಇತ್ತು, Illon colony ಇತ್ತು. So ಇಲ್ಲಿ ನೋಡಿದ್ರೆ students, ಈ colony ಬಂದ್ಬಿಟ್ಟು this was called, okay, so these two colonies led to the formation of two different strains, okay, it led to the formation of two different strains. What was the strain? One was called as the R strain, one was called the S strain. One was called the R strain, one was called the S strain. Okay, so R strain and S strain. Ill nodi S strain banbit. Ma'am, what is the full form of S andre students? S strain is called as the smooth strain. S strain is called as what? Smooth strain. Okay. This one, R strain, is called the rough strain. Okay. Rough strain. ಇವಾಗ ನಮ್ಮ ಮನೆಯಲ್ಲೂ ಲೆಟ್ಸ್ ಸೇ ಎರಡು ಸಿಬ್ಲಿಂಗ್ಸ್ ಇರುತ್ತೆ. One will be very soft, like soft spoken, will not be angry. One will be very rough and tough. Hey, I don't want to talk to you, go man. ಆ ಥರ ಒಂದು ರೂಡ್ ಬಿಹೇವಿಯರ್ ಇರುತ್ತಲ್ವಾ, ಆ ಥರನೇ ಇಲ್ಲಿ ಒಂದು ಸ್ಮೂತ್ ಸ್ಟ್ರೈನು ಇದೆ, ರಫ್ ಸ್ಟ್ರೈನು ಇದೆ. ಬಟ್ ಅವ್ರ ಸ್ಟ್ರಕ್ಚರ್ ಬೇಸಸ್ ಯಾಕೆ ಹೆಂಗಿರುತ್ತಂತ ಇಲ್ಲಿ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡ್ತೀನಿ. ಇಲ್ಲಿ ನೋಡಿ, ಸ್ಮೂತ್ ಸ್ಟ್ರೈನಲ್ಲಿ ಸ್ಟೂಡೆಂಟ್ಸ್ ಇಲ್ಲೊಂದು ಕ್ಯಾಪ್ಸ್ಯೂಲ್ ಫಾರ್ಮೇಷನ್ ಇರುತ್ತೆ. ಒಂದು ಮ್ಯೂಕೋಪಾಲಿಸ್ಯಾಕರೈಡ್ ಕ್ಯಾಪ್ಸ್ಯೂಲ್ ಫಾರ್ಮೇಷನ್ ಇದೆ. That is leading to this becoming the smooth strain. This becoming to the smooth strain. So there will be a, you can say a mucus coating, you can say a capsule which is made up of mucopolysaccharide. So it is formed by, it's formed with mucopolysaccharide. Mucopolysaccharide. On capsule tara, okay, on the outer layer tara. Okay, ಆ ಒಂದು ಲೇಯರ್ ಇರುತ್ತೆ, ವಿಚ್ ಇಸ್ ಬಿಕಾಸ್ ಆಫ್ ವಿಚ್ ದಿಸ್ ವಿಲ್ ಬಿ ಸ್ಮೂತ್ ಸ್ಟ್ರೈನ್. ಆ ಲೇಯರ್ ಆರ್ ಸ್ಟ್ರೈನಲ್ಲಿ ಆಬ್ಸೆಂಟ್ ಆಗಿರುತ್ತೆ. ಆ ಲೇಯರ್ ಏನಾಗುತ್ತೆ, ಆರ್ ಸ್ಟ್ರೈನಲ್ಲಿ ಆಬ್ಸೆಂಟ್ ಆಗಿರುತ್ತೆ. ಸೊ ಆರ್ ಸ್ಟ್ರೈನ್ ಬಂದ್ಬಿಟ್ಟು ಸಮಥಿಂಗ್ ಲೈಕ್ ದಿಸ್ ಇಟ್ ವಿಲ್ ನಾಟ್ ಬಿ ಸ್ಮೂತ್. It will not be smooth. It will be different. Okay, it will be rough. Okay, rough. So it will be rough. They will not have the capsule layer. No capsule layer. So this is going to be rough. That is going to be smooth. Clear to all of you? Is this clear? Done? Okay, yes. Very good. Now students, like when Griffith was working on this experiment, he understood one thing. He understood that this S strain, no, smooth strain, S strain is virulent. Virulent andre eno? Ma'am, disease causing, ma'am. Virulent andre eno? Disease causing. So S strain was virulent. Virulent andre disease causing. Okay. Disease causing. Now R strain andre eno? R strain, obviously S strain is virulent, means R strain is non-virulent. Non-virulent, and it was not disease causing. So some of them told that yes, S strain is virulent, illandre disease causing because of the mucopolysaccharide or because of the mucus coating. Because of that only maybe this S strain is actually virulent, andre disease causing. On capsule layer R strain absent agito. So everyone were like, it is absent. So adikke mostly R strain is not virulent. Okay, iter on conclusion ito. Now keeping this in mind, Griffith did an experiment on mice. Okay, Griffith did experiment on what, students? He did it on mice. So what was the experiment setup heltini? So first I will draw beautiful mice here. Okay, mice number 1, tail. Okay, mice number 2, mice number 3, mice number 4. Okay, 4 mice are there, right? So first we will start. Okay, first we will start. Yes, Anita, is everyone smashing the like button? Attendance mark ಮಾಡ್ತಾ ಇದ್ದಾರಾ? ಚೆಕ್ ಮಾಡಿ ಬೇಗ. ಚೆಕ್ ಮಾಡಿ ನನಗೆ ಹೇಳಿ ಅಟೆಂಡೆನ್ಸ್ ಮಾರ್ಕ್ ಮಾಡ್ತಾ ಇದ್ದಾರಾ ಇಲ್ವಾ ಅಂತ. ಓಕೆ, ಎಸ್. ಸೊ ಇಲ್ಲಿ ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ಇಲ್ಲೇನಾಯಿತು, ಫಸ್ಟ್ ನಾವು ಮೈಸ್ನ ಎಸ್ಟ್ರೇನ್ ಇಂದ, ಓಕೆ, ಮೈಸ್ ವಿತ್ ಎಸ್ಟ್ರೇನ್, ವಿ ಇಂಜೆಕ್ಟೆಡ್ ಎಸ್ಟ್ರೇನ್ ಇನ್ ಟು ದ ಮೈಸ್. ಓಕೆ, ಮೈಸ್ ವಾಸ್ ದೇರ್. ಸೊ ಇದು ಮೈಸ್ ಅಂತ ಅರ್ಥ ಆಗುತ್ತಲ್ವಾ? ಡೋಂಟ್ ಟೆಲ್ ಮಿ ಮ್ಯಾಮ್, ವಿ ಕಾಂಟ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದಿಸ್ ಇಸ್ ಮೈಸ್. Okay, mice 1, mice 2, mice 3, mice 4. Okay, so mice we injected with S strain. So illena ma'am, obvious ma'am, what will happen? Illi mice is going to die. Mice died. Students, mice will die, no? I am using a pathogenic, a disease-causing person or disease-causing strain and I am injecting into the mice. So, obviously, mice is going to die. Adikeno doubt illa. Ma'am, nanu gottu ma'am, strain virulent agittu, disease-causing agittu. Obviously, mice will inject madre, mice is going to die. Second one also is obvious because second one was our R strain. Okay. So, we injected, what happened? The mice is alive. Mice is alive. Why? Because R strain is non-virulent. He is power. Okay. Now, here we did a combination. Okay. Here we did what? A combination. We did, okay, this fellow is very, very virulent. ಏನು ಮಾಡೋಣ ನಮಗೆ ಅದನ್ನ heat ಮಾಡಿಬಿಟ್ಟು, so we will do heat killed S strain. Heat killed S strain. ಇಲ್ಲೇನು ಎಕ್ಸ್ಪೆಕ್ಟ್ ಮಾಡ್ತಾ ಇದ್ದೀರಾ ಮ್ಯಾಮ್ ಹೀಟ್ ಯೂಸ್ ಮಾಡಿದ್ದೀರಾ ಸೊ ಆಬ್ವಿಯಸ್ಲಿ ಮೈಸ್ ವಿಲ್ ಬಿ ಅಲೈವ್ ಓಕೆ ವೆರಿ ಗುಡ್ ಎಸ್ ವೆರಿ ಗುಡ್ ಸೊ ಇಲ್ಲಿ ಮೈಸ್ ವಾಸ್ ಅಲೈವ್ ಬಿಕಾಸ್ ಇಲ್ಲಿ ನೀವು ಹೀಟ್ ಯೂಸ್ ಮಾಡಿದ್ದೀರಾ ಈ ಆಕ್ಟಿವ್ ಸಬ್ಸ್ಟೆನ್ಸ್ನ ಇನ್ಆಕ್ಟಿವ್ ಮಾಡಿದ್ದೀರಾ ಬಿಕಾಸ್ ವೆನ್ ಯು ಗಿವ್ ಹೀಟ್ ಮೋಸ್ಟ್ ಆಫ್ ದ ಪ್ಯಾಥೋಜನ್ಸ್ ವಿಲ್ ಡೈ ಅಲ್ವಾ ಸೊ ಆ ಥರ ಹೀಟ್ ಕಿಲ್ಡ್ ಎಸ್ ಸ್ಟ್ರೇನ್ ದ ಮೈಸ್ ಇಸ್ ಅಲೈವ್ ಇವಾಗ ಇನ್ನೊಂದು ಕಾಂಬಿನೇಷನ್ ಸೊ ಗ್ರಿಫಿತ್ ವಾಸ್ ಲೈಕ್ ಓಕೆ ನಾನು ಇಷ್ಟು ಮಾಡಿದ್ದೀನಿ ಲೆಟ್ ಮಿ ಡೂ ದಿಸ್ ಟು ಕಾಂಬಿನೇಷನ್ both the scenarios where the mice is alive alva both the scenarios where the mice is alive let me mix that okay let me mix that so what did he do students illi on different combination try madidare he used heat killed S strain plus R strain and ನೀವಿಲ್ಲಿ ಹೇಳಿ ಏನಾಗುತ್ತೆ ಅಂತ what do you expect here what would happen here just give a guess ಮಾಡಿದ್ರೇನು ಪ್ರಾಬ್ಲಮ್ ಇಲ್ಲ but just tell me what do you think would have happened here come on any idea what happened any idea what happened ಇಲ್ಲಿ ನಾರ್ಮಲ್ ಆಗಿ ಥಿಂಕ್ ಮಾಡಿ ಮ್ಯಾಮ್ ನಾನು ಗ್ರಿಫಿತ್ ಆದರೆ I would think that mice would be alive because heat kill allu mice alive agittu R strain individually inject mado time allu mice alive agittu so it's obvious that the mice is going to be alive but what happened twist happened ill twist enayt students the mice died okay the mice dies why because the whole conversation happens because from the heat kill red al bardre ಕಾಣಿಸ್ತಾ ಇದೆಯಾ yes okay from the entire heat killed S strain okay heat killed S strain okay to R strain heat killed S strain to R strain something moved let's say example students ನಿಮಗೊಂದು ಫ್ರೆಂಡ್ ಇದೆ okay ತುಂಬ ನೆಗೆಟಿವ್ ಮಾತಾಡ್ತಾನೆ ಫುಲ್ ಟೈಮ್ ಏನು ಬಗ್ಗೆ ಕೇಳಿದ್ರೇನು ಫಸ್ಟ್ ನೆಗೆಟಿವ್ ಬರೋದು ಅವನ ಫ್ರಮ್ ಹಿಸ್ ಮೌತ್ ಓಕೆ ಅವನ ಬಾಯಿಂದ ಫಸ್ಟ್ ಬರೋದೇ ನೆಗೆಟಿವ್ ಲೆಟ್ಸ್ ಸೇ 1 ಮಂತಲ್ಲಿ ನಿಮಗೆ ಅದು ಐಡೆಂಟಿಫೈ ಆಗಲ್ಲ ಸೆಕೆಂಡ್ ಮಂತಿಂದ ನೀವು ಆ ಥರ ಮಾತಾಡೋಕ್ಕೆ ಶುರು ಮಾಡ್ತೀರಾ ಸ್ಲೋಲಿ 10% ಶುರು ಮಾಡ್ತೀರಾ ಏನು ಹೇಳಿದ್ರೇನೋ ಫಸ್ಟ್ ನೆಗೆಟಿವ್ ಬರುತ್ತೆ ದೆನ್ ಯು ವಿಲ್ ರಿಯಲೈಸ್ ಅಯ್ಯೋ ವೈ ಆಮ್ ಐ ಟೆಲಿಂಗ್ ನೆಗೆಟಿವ್ ಐ ಶುಡ್ ಸ್ಟಾರ್ಟ್ ಟೆಲಿಂಗ್ ಪಾಸಿಟಿವ್ ಆಫ್ಟರ್ 5 months then 10 months then 1 year down the lane you will also become just like him alva aa tarane illi heat kill S strain bandbit villain he is the villain of the story R strain is the hero of the story but heat kill S strain inda eno ond element allinda R strain ige move agide because it moved R strain became the villain R strain became virulent so from the heat kill S strain something transformed okay what is the word that you use here something transformed and that is called the transforming principle That is called what? The transforming principle. Artha aita? So something got transformed from the heat killed S strain to R strain. Students, idu tumba common. It's tumba common because bacteria dalli sexual reproduction, biological classification I have taught you, sexual reproduction concept ಬಗ್ಗೆ ಮಾತಾಡೋ ಟೈಮಲ್ಲಿ they tell that bacteria does 3 types of sexual reproduction: conjugation, transformation, transduction. ಅದರಲ್ಲಿ ಸೆಕೆಂಡ್ ಸ್ಟೆಪ್ ಏನು? ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಷನ್. ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಷನ್ ಅಂದರೆ ಲೆಟ್ಸ್ ಸೇ ಎಲ್ಲಾದರೂ ಒಂದು ನೇಕೆಡ್ ಡಿ ಎನ್ ಎ, ಓಕೆ, ನೇಕೆಡ್ ಡಿ ಎನ್ ಎ ಅಂದರೆ ವಿತೌಟ್ ದ ಸೆಲ್ ಆ್ಯಂಡ್ ಎನಿಥಿಂಗ್, ನೇಕೆಡ್ ಡಿ ಎನ್ ಎ ಇಸ್ ಪ್ರೆಸೆಂಟ್, ಇಟ್ ವಿಲ್ ಬಿ ಪಿಕ್ಡ್ ಅಪ್ ಬೈ ದ ಬ್ಯಾಕ್ಟೀರಿಯಾ. Streptococcus pneumonia is a bacteria. It's a bacterium. Alva. It's a bacterial disease. So this naked DNA from the S strain would have been picked up by the R strain, making R strain virulent. Namagi full story gotu, but adre kuda with experiments only we can prove. Namak just to, let's say, I cannot just say things in science. I need to prove it with experiments. If I only prove it with experiments, it makes sense. Okay, it makes sense. So this is clear. So this is what happened. Okay, now next set of people came. Yes, transfer of genetic material. Yes. So students, he called this, okay, he called this the transforming principle, and experiment also was called transforming principle. He did not go further with the experiment. He was like, I don't know what it was, but I will just call it the transforming principle anta. Illi nodidre there is another people called as Avery. Okay, Avery, MacLeod, McCarty. Avery, MacLeod and McCarty. These 3 people were like, okay, wait a minute. I know something that I can do, okay? Something that I can do, that let's try what is the biochemical nature. So, this combination itilva, heat killed S strain, heat killed S strain plus R strain. So they were checking the biochemical nature of this heat killed S strain, that what would have got transformed from heat killed S strain to R strain. So they did an experiment, students. They did an experiment, and what did they do? So look here, 1, 2 and 3. They took 3 setups, okay? They took 3 setups, and in this 3 setups ಏನಾಯಿತು? So our confusion ಏನಂದ್ರೆ, our confirmation ಇದ್ದಿಲ್ಲ that DNA is the genetic material ಅಂತ. Our confusion ಇತ್ತು, what if it is protein? What if it is protein? Okay, some of them had confusion, what if it is RNA? And some of them had confusion, what if it is DNA? So Avery, MacLeod and McCarty experiment mada time alli, they were like, okay, namagond idea try madana, let's degrade these people. Degrade andre, let's just break them down and let's digest them, okay, with the help of enzymes. With the help of what, students? With the help of enzymes. So that enzymes were what? The enzymes were protease, RNase, DNase. Protease, RNase and DNase. Okay. So these enzymes were going to degrade. These enzymes were going to digest these people to just do the experiment. So ಇಲ್ಲಿ ನೋಡಿ experiment ಗೆ ಯಾವ ಕಲರ್ ಯೂಸ್ ಮಾಡಬೇಕು? ಪ್ರೋಟೀನ್ ಗೆ ಗ್ರೀನ್ ಯೂಸ್ ಮಾಡೋಣ. ಸೊ ಇದು ಪ್ರೋಟೀನ್, ಇಲ್ಲೂ ಪ್ರೋಟೀನ್, ಇಲ್ಲೂ ಪ್ರೋಟೀನ್. ಓಕೆ, ಅದಾದಮೇಲೆ ಬ್ಲೂ ಯೂಸ್ ಮಾಡೋಣ RNA ಗೆ. ಪ್ಲೀಸ್ ರಿಮೆಂಬರ್ ದ ಕಲರ್. Blue for RNA, blue for RNA. DNA again use madana, pink use madana, DNA, DNA, DNA. Okay, so this happened, yes or no? This happened. So what are they going to do here? They used protease enzyme. They used what enzyme? Protease enzyme. Students, please notes alla download ಮಾಡಿ Vedantu Karnataka Telegram channel ಇಂದ. Okay, don't forget, okay? ಇದನ್ನ ಶಿಫ್ಟ್ ಮಾಡಬೇಕಾಗಿತ್ತು. So protease, first we are going to use protease. Protease alli ಏನು students ಇಲ್ಲಿ digest ಆಗೋದು? Protein digest ಆಗುತ್ತಾ, RNA digest ಆಗುತ್ತಾ, DNA digest ಆಗುತ್ತಾ? ಬೇಗ chat box ಅಲ್ಲಿ ತಿಳಿಸಿ. ಬೇಗ. Who is going to get degraded here? Who is going to get degraded here? Please tell me in the chats. Is it going to be RNA? Is it going to be DNA? Is it going to be who? Huh? It's going to be who? DNA, RNA or protein? Come on, all of you. It's going to be definitely protein because I have used protease. It's an enzyme that would degrade protein. Okay? How to download notes? Last I'll toasteni. Okay? At the end of the video, I will show you. Okay? So protease done. So ill students ಏನಾಗುತ್ತೆ? Proteins are going to get digested alva. So when proteins are getting digested, see, ಈ experiment ಬಂದ್ಬಿಟ್ಟು ಒಂದು small problem ಇದೆ. ಈ experiment NCERT ಅಲ್ಲಿ ಹೋದ್ರೆ ನಿಮಗೆ ಏನು ಅರ್ಥ ಆಗಲ್ಲ. ಸೊ ಇಲ್ಲಿ ನಾನು ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಷನ್ ಅಂತ ಹೇಳಿ ಕನ್ಫ್ಯೂಸ್ ಮಾಡಲ್ಲ. ಜಸ್ಟ್ ನಾರ್ಮಲ್ ಆಗಿ ಲೆಟ್ಸ್ ಸೇ ಇಫ್ ಐ ವಾಸ್ ಡೂಯಿಂಗ್ ಗ್ರಿಫಿತ್ಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ವಾಟ್ ವುಡ್ ಹ್ಯಾವ್ ಹ್ಯಾಪನ್. ಸೊ ಇಲ್ಲಿ ಪ್ರೋಟಿಯೇಸ್ ಯೂಸ್ ಮಾಡಿಬಿಟ್ಟು ಪ್ರೋಟೀನ್, ಓಕೆ ಒನ್ ಸೆಕೆಂಡ್, ಪ್ರೋಟಿಯೇಸ್ ಯೂಸ್ ಮಾಡಿಬಿಟ್ಟು ಹಿಯರ್ ಪ್ರೋಟಿಯೇಸ್ ವಿ ಯೂಸ್ಡ್ ಆ್ಯಂಡ್ ದ ಪ್ರೋಟೀನ್ ಗಾಟ್ ಡೈಜೆಸ್ಟೆಡ್. ಇವಾಗ RNA ಮತ್ತು DNA ಇದೆ ಅಲ್ವಾ. ಸೊ ವೆನ್ ಯು ಇಂಜೆಕ್ಟೆಡ್ ಇನ್ ಟು ದ ಮೈಸ್ ಯು ವಿಲ್ ನೋಟಿಸ್ ದಟ್ ವಾಟ್, ಯು ವಿಲ್ ನೋಟಿಸ್ ದಟ್ ದ ಮೈಸ್ ಇಸ್ ಸ್ಟಿಲ್ ಡೈಯಿಂಗ್, ರೈಟ್? ದ ಮೈಸ್ ವಿಲ್ ಡೈ. Mice dies. That means here transformation has happened. Okay? Telegram ಅಲ್ಲಿ ಇವಾಗ ನನ್ನೇ ನೋಟ್ಸ್ ಪೋಸ್ಟ್ ಆಗಿಲ್ಲ, ಪೋಸ್ಟ್ ಮಾಡ್ತೀನಿ. ಓಕೆ, ಪೋಸ್ಟ್ ಮಾಡ್ತೀನಿ. ಯೂಶಲಿ ನಾವು ಸೆಷನ್ PDF ಅಲ್ಲಿ ಸೆಷನ್ ಇದು ಡಿಸ್ಕ್ರಿಪ್ಷನ್ ಬಾಕ್ಸಲ್ಲೇ PDF ಎಂಟರ್ ಮಾಡ್ತೀವಿ ಬಿಕಾಸ್ ಇಟ್ಸ್ ವೆರಿ ಈಸಿ ಫಾರ್ ಯು ಪೀಪಲ್ ಟು ಡೌನ್ಲೋಡ್ ದೆನ್ ಆ್ಯಂಡ್ ದೇರ್. ಓಕೆ, ಬಿಕಾಸ್ ಟೆಲಿಗ್ರಾಮಲ್ಲಿ ಸಮ್ಟೈಮ್ಸ್ ಸಮ್ ಆಫ್ ಯು ವಿಲ್ ಜಾಯಿನ್ ಲೇಟ್, ಇಲ್ಲಾಂದರೆ ಫೈಲ್ಸಲ್ಲಿ ಹೋಗಿ ನೋಡಲ್ಲ ಕರೆಕ್ಟಾಗಿ, ನೇಮ್ ಕರೆಕ್ಟ್ ಆಗಿರಲ್ಲ, ಆ ಥರ ತುಂಬ ಪ್ರಾಬ್ಲಮ್ಸ್ ಇರುತ್ತೆ. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ ಸ್ಟೂಡೆಂಟ್ಸ್, ಮೈಸ್ ಡೈಸ್ ಅಂದರೆ ಹಿಯರ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಷನ್ ಇಸ್ ಹ್ಯಾಪನಿಂಗ್. ಓಕೆ, ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಷನ್ is happening. Transformation is happening. Okay, so we understood one thing. Okay, if the mice is dying, that means protein is not the genetic material, protein is not the transforming principle anta. Illen use ಮಾಡ್ತೀವಿ? Here we are going to use RNAs. Here we are going to use what? We are going to use RNAs. So RNAs use ಮಾಡೋ ಟೈಮಲ್ಲಿ ನಮಗೆ ಆಲ್ರೆಡಿ ಅರ್ಥ ಆಗುತ್ತೆ that okay, RNAs use ಮಾಡಿದ್ರೆ RNA degrade ಆಗುತ್ತೆ ಅಲ್ವಾ. RNAs ಯೂಸ್ ಮಾಡಿದ್ರೆ RNA ಡಿಗ್ರೇಡ್ ಆಗುತ್ತೆ. ಸೊ ಇಫ್ RNA is going to get degraded, let's say RNA is the genetic material, then what will be the scenario? ಸೊ ಇಲ್ಲಿ ನೋಡಿದ್ರೆ ಸ್ಟೂಡೆಂಟ್ಸ್ ಏನಾಗುತ್ತೆ, ಇಲ್ಲೂ ಸೇಮ್ ಥಿಂಗ್, ದ ಮೈಸ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಡೈ, ಮೈಸ್ ಡೈಸ್. ಓಕೆ, ಸೊ ಅಂದರೆ ಇಲ್ಲೂ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಷನ್ ನಡೆಯುತ್ತೆ. ಹಿಯರ್ ಆಲ್ಸೋ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಷನ್ ವಿಲ್ ಹ್ಯಾಪನ್. ಓಕೆ, ಹಿಯರ್ ಆಲ್ಸೋ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೇಷನ್ ವಿಲ್ ಹ್ಯಾಪನ್. So that means we understood two things now. Protein alla, it's not RNA. Now last try bandu DNA. Okay, ee tara ishtu clear agi nim NCERT alli explain madilla. I am explaining so you understand. Okay, ಇಲ್ಲಿ ನೋಡಿ, here we are going to use DNAs. Here we are going to use DNAs. DNAs use ಮಾಡೋ ಟೈಮಲ್ಲಿ what are we expecting? ಕೆಳಗೆ ಬರೀತೀನಿ. What are we expecting? The mice is going to live. Why? Because the mice will live because we have digested the DNA. Okay, digested the DNA. So here the mice lives. That means no transformation. No transformation. No transformation. So here what do we understand, students? Protein is not the one. DNA, RNA is not the one. It is DNA who has been transformed. So illinda namagondu conclusion draw agutte that transforming principle is who? It is because of your DNA. But idu explain ಮಾಡಕ್ಕೆ ಅವರು ಯೂಸ್ ಮಾಡಿರೋ ಮೆಥಡ್ ಅಷ್ಟು ಜಸ್ಟಿಫೈಡ್ ಅಲ್ಲ. ಈ ಎಕ್ಸ್ಪೆರಿಮೆಂಟಿಗೆ ಎಲ್ಲೋ ಒಂದು ಪ್ರಾಬ್ಲಮ್ ಇತ್ತು, ದಟ್ ಸಮಥಿಂಗ್ ಕ್ಲಾರಿಟಿ ಪ್ರಾಬ್ಲಮ್ ಇತ್ತು. ಸೊ ವಾಟ್ ಹ್ಯಾಪನ್ ಸ್ಟೂಡೆಂಟ್ಸ್? ಡಿಡ್ ಯು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದಿಸ್ ಎವ್ರಿವನ್? Quickly tell me in the chat box, did you all understand this? Anyone has any doubts? ಡೌಟ್ ಇದ್ದರೆ ಆವಾಗಲೇ ಕೇಳಬೇಕು. ಓಕೆ, ನನ್ನ ಕ್ಲಾಸಲ್ಲಿ ಮ್ಯಾಮ್ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸಲ್ಲಿ ಕೇಳ್ತೀನಿ ಮ್ಯಾಮ್, ನಾನು ತುಂಬ ಶೈ ಆಗಿದ್ದೀನಿ ಯೂಟ್ಯೂಬಲ್ಲಿ ಡೌಟ್ ಕೇಳೋಕ್ಕೆ, ಆ ಥರ ಯು ಶುಡಂಟ್ ಹ್ಯಾವ್ ಎನಿ ಕಾಂಪ್ಲೆಕ್ಸ್. ಓಕೆ, ಡೈರೆಕ್ಟ್ಲಿ ಯು ಶುಡ್ ಆಸ್ಕ್ ಮೀ ಡೌಟ್. ವಿ ಆರ್ ಡೂಯಿಂಗ್ ದಿಸ್ ಫಾರ್ ಯು. ಓಕೆ, ಸೊ ವಿ ವಾಂಟ್ ಆರ್ ಕರ್ನಾಟಕ ಸ್ಟೂಡೆಂಟ್ಸ್ ಟು ಕಮ್ ಅಪ್ ಇನ್ be it in your MBBS, be it in your KCET. So ಅದಕ್ಕೆ KCET ಕ್ಲಾಸ್ ನಿಮಗೆ PUCಗೂ ಯೂಸ್ ಆಗುತ್ತೆ, ಸೇಮ್ ಕ್ಲಾಸೇ. ಓಕೆ, ಸಮ್ ಕ್ವೆಶನ್ಸ್ ವಿಲ್ ಬಿ ಸಾಲ್ವಿಂಗ್ ದೆರ್. ಓಕೆ, ನೋಟ್ಸ್ ನೀವು ಇಲ್ಲಿ ಓಕೆ ಅರ್ಥ ಆಯಿತು ಅನಿತಾ. ಈ ನೋಟ್ಸ್ ನಾನು ಸೇವ್ ಮಾಡಿಬಿಟ್ಟು ಈ ಕ್ಲಾಸ್ ಆದಮೇಲೆ ಒಂದು ಹಾಫ್ ಆನ್ ಅವರ್ ಇಲ್ಲಾಂದರೆ ಒಂದು 15 ಮಿನಿಟ್ಸ್ ಆದಮೇಲೆ ಡಿಸ್ಕ್ರಿಪ್ಷನ್ ಬಾಕ್ಸಲ್ಲಿ ಇರುತ್ತೆ. ಓಕೆ, ಬಿಕಾಸ್ ಈ ನೋಟ್ಸ್ ಇವಾಗ ಬರೀತಾ ಇದ್ದೀನಿ ಅಲ್ವಾ, ಎಂಟಿ ಸ್ಲೈಡ್ಸ್ ಎತ್ಕೊಂಡು ಬಂದಿದ್ದೀನಿ. ಸೊ ಕಂಪ್ಲೀಟಾಗಿ ಆಫ್ಟರ್ ಐ ಫಿನಿಶ್ ರೈಟಿಂಗ್, ಸೊ ಅದಾದಮೇಲೆ ನಿಮಗೆ ನೋಟ್ಸ್ ಎಲ್ಲ ಡಿಸ್ಕ್ರಿಪ್ಷನ್ ಬಾಕ್ಸಲ್ಲಿ ಇರುತ್ತೆ. ಓಕೆ, ಬೈ ಚೇತನ್, ನೋಟ್ಸ್ ಸಿಕ್ಕಲ್ಲ? ಯು ವಿಲ್ ಗೆಟ್ ದ ನೋಟ್ಸ್. ಐ ಶೋ ಯು ಹಿಯರ್ ಓನ್ಲಿ. ವೈ ವಿಲ್ ಯು ನಾಟ್ ಗೆಟ್ ದ ನೋಟ್ಸ್? ವಾಟ್ ವಿಲ್ ಐ ಡೂ ವಿತ್ ಮೈ ನೋಟ್ಸ್? ಇಲ್ಲಿ ನೋಡಿ. ಓಕೆ, ಹಿಯರ್ go to the channel, go to live. Okay, ನೋಡಿ ಇಲ್ಲಿ ಸೆಷನ್ PDF ಲಿಂಕ್ ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. ಓಕೆ, ನಾನು ಇದು ಮೇಲೆ ಹಾಕ್ಸ್ತೀನಿ, ಸೊ ಡೋಂಟ್ ವರಿ, ಇಲ್ಲೇ ಇರುತ್ತೆ ಸೆಷನ್ PDF ಲಿಂಕ್ ಅಂತ. ಇದಲ್ಲಿ ಕ್ಲಿಕ್ ಮಾಡಿ ಫೋನ್ ನಂಬರ್ ಎಂಟರ್ ಮಾಡಿ, ಇಲ್ಲಾಂದರೆ ಇಮೇಲ್ ID ಅಂತ ಎಂಟರ್ ಮಾಡಿ, ಆಟೋಮ್ಯಾಟಿಕ್ಲಿ ನೋಟ್ಸ್ ನಿಮಗೆ ಓಪನ್ ಆಗುತ್ತೆ. Okay, automatically notes ನಿಮಗೆ ಓಪನ್ ಆಗುತ್ತೆ. Understood? Why you won't get the notes? What will we do with the notes? Okay, don't spread rumors, students. I don't like it. Okay, rumors spread ಮಾಡಬೇಡಿ. Teachers are trying very hard to do whatever is possible. So unnecessary rumors is not good. Okay, ನೋಡಿ ಇಲ್ಲಿ KCT ಕ್ಲಾಸ್ದು ಇಲ್ಲಿ ನಡೀತಾ ಇದೆ. Okay, so here session PDF ಲಿಂಕ್ ಅಪ್ಡೇಟ್ ಮಾಡಬೇಕು, ಅಪ್ಡೇಟ್ ಮಾಡೋಕ್ಕೆ ಹೇಳ್ತೀನಿ. ಓಕೆ, ಇಲ್ಲ ಅಂದರೆ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್ ಹೋಗಿ ಡೈರೆಕ್ಟ್ ಆಗಿ. So you go to the Telegram channel, view in Telegram, download ಮಾಡಬೇಕು, ಅದಾಗಲ್ಲ. So in Telegram we will be sharing the notes. Clear? I'll tell teachers to share the notes then and there. You don't worry about it. Okay, it's not your headache, it's my headache. So I'll do that. I'll take care of it. Okay, done. Yes, today my notes you will get then and there. Okay, my notes within 15 minutes you will get it updated in the description box. Okay, all the NEET notes, because just one class got over, so that class notes will be there. Okay, now is this clear? So in the biochemical characteristics, okay, biochemical characteristics of transforming principle, we understood what, students? We understood that the transforming principle, okay, the transforming principle is DNA. Okay, it was DNA, but not all agreed. Okay, but not all agreed, because of which what happened? Because of which we had to do Hershey and Chase experiment. Ready, all of you? Okay, I want just like 2 minutes. I'll just refill water and cup. Okay, so 5 minutes break not required. Okay, 6:12, just 2 minutes, students. Okay, just be here, don't run away, don't run away. Hello. Thank you for waiting, students. Okay. Ma'am keeps forgetting her water bottle. So, what to do? Problems. Problems after problems. Okay, come on. So, Hershey and Chase in 1952. Okay, they did an experiment. Now, what and all they took? First, let's see the materials. Alva? Inalla materials use ಮಾಡಿ ಅವ್ರಿಗೆ experiment ಮಾಡಿದ್ದಾರೆ ಅಂತ. Hershey and Chase actually, they uni-equally. Okay. Equivocal. EQ. Then. Proof that DNA is the genetic material. Andre ultimate proof that DNA is the genetic material anta prove ಮಾಡಿದ್ದಾರೆ. So ಇಲ್ಲಿ ನೋಡಿದ್ರೆ Hershey and Chase ಏನಲ್ಲ setup use ಮಾಡಿದ್ದಾರೆ ಅಂತ important ನಿಮಗೆ. So Hershey and Chase use ಮಾಡಿರೋದು first they used a bacteriophage. Now what is a bacteriophage? This is why your 11th topic is important for you to understand. Okay, bacteriophage is a virus that infects bacteria. It is a virus that infects bacteria. Okay, bacteriophage is what? A virus that infects bacteria. Virus that infects bacteria. Now they use that. Okay, one was bacteriophage. Okay, next thing that they used, students, was an E. coli cell. Okay, an E. coli. They used an E. coli to infect. Okay, E. coli will act as the host. Okay, E. coli will act as the host. E diagram ilva? This diagram same thing I am showing you, but I am drawing and showing you here. Okay, they used E. coli. So this was the first material setup. This was the second setup. Okay, now they use three main steps. Okay, in that the most important step, students, is called as radioactive labeling. What is it called? Radioactive labeling. Radioactive labeling andre ನಮಗೆ ಲೆಟ್ಸ್ ಸೇ ನಾವು ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡೋ ಟೈಮಲ್ಲಿ ಸೆಂಟ್ರಿಫಿಕೇಶನ್ ಮಾಡೋ ಟೈಮಲ್ಲಿ ಇಲ್ಲಾಂದರೆ ಏನಾದರೂ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡೋ ಟೈಮಲ್ಲಿ ನಮಗೆ ಪೆನ್ನಿಂದ ಮಾರ್ಕರಿಂದ ಲೇಬಲ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. ಇಫ್ ಐ ವಾಂಟ್ ಟು ನೋ ವಾಟ್ ಈಸ್ ಎಕ್ಸಾಕ್ಟ್ಲಿ ಇನ್ಸೈಡ್ ಇಟ್, ಐ ಹ್ಯಾವ್ ಟು ಡೂ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಲೇಬಲಿಂಗ್. I have to use radioactive probes to see it under auto radiography. Andre I want to see the highlight. So adanna highlight ಮಾಡಬೇಕಂದ್ರೆ ನಾನು ಅದನ್ನ ಲೇಬಲ್ ಮಾಡಬೇಕು. So label ಮಾಡಬೇಕಂದ್ರೆ I do radioactive labeling. So radioactive labeling ಮಾಡಕ್ಕೆ they have taken two things, students. What was the one thing that they took? They took P35, okay, sorry, P32 and S35, sulfur 35 ಮತ್ತೆ phosphorus 32, P32 and sulfur 35. Ivaga phosphorus, we know it is only present in DNA. So only DNA will get radioactively labeled. We know sulfur is only in protein, so only protein will get labeled. E on concept alli radioactive labeling was designed. Okay, radioactive labeling was designed. Understood? So they took only DNA, only protein. So we understood that, okay, if I use P32, only my DNA is going to be labeled. If I take S35, only my protein is going to be labeled. E on concept alli, let's say P32. Okay, we grow the medium. Okay, the medium is P32. We grow the bacteriophage there. What will happen, students? So we'll have our bacteriophage. Okay, bacteriophage is there. Now DNA will get radioactively labeled. Okay, DNA will get radioactively labeled. Correct, alva? Let's say I am growing this bacteriophage on S35 protein. So here what is going to happen? The protein is going to get radioactively labeled. Protein is going to get radioactively labeled. Done. This is the simple concept. Okay, now let's see here. Illi brightness ಕಮ್ಮಿ ಮಾಡಬೇಕು. ಕಾಣಿಸ್ತಾ ಇದೆಯಾ students? Is it visible? Yeah, it's almost visible. I will not be visible that much, okay, because light ತುಂಬ ಕಡಿಮೆ ಇದೆ ಇಲ್ಲಿ. One second. Okay, I don't know now if it is better. Okay. Okay. Now, look here. Illi, bacteriophage is there this side. Can you see bacteriophage? Radioactive S35 labeled protein capsule. So, S35 use madi. Okay, S35 use madi. Now, protein na label madi. Now, this side, whatever is happening with the radioactively labeled protein is happening. This side, whatever is happening with DNA is happening. Okay. So, happening is happening. Okay. So, illi enagutte. First step banbit students, infection. First step enirutte infection. Infection andre where I am using the bacteriophage to infect the E. coli. Because bacteriophage is what? Virus that infects bacteria. What is a E. coli? It's a bacteria. So, connect ಆಗ್ತಾ ಇದೆಯಾ? Bacteriophage use ಮಾಡಿ ನಾನು E. coli ನ infect ಮಾಡ್ತೀನಿ. So, first step ಏನು? Infection. Because infect ಮಾಡಿದ್ರೇನೆ ನನಗೆ ಅರ್ಥ ಆಗೋದು, okay, there is transformation happening and whoever is getting radioactively labeled will be present inside the cell ಅಂತ. So, ಇಲ್ಲಿ ನೋಡಿದ್ರೆ infection ಆಗುತ್ತೆ. So, here the bacteriophage is going to infect the E. coli. E. coli got infected. E. coli got infected. Now, second step, what is happening, students? After the E. coli gets infected, see, what is the job of virus? Virus will release the genetic material into the host. That is their main job, alva. So, adamele blending process in the using a mixer blender. Okay, using a mixer blender, we will be removing the protein coat. We will be doing what? We will be removing the protein coat. A protein coat remove madadmele, the genetic material will be inside the DNA. After that, what is going to happen with the process of centrifugation? With the process of what? Centrifugation, we will be checking. Okay, let's say if at the end of centrifugation, what we are going to get? So, this is your centrifugation tube. Okay, this is your centrifugation tube, and here let's say a heavy substance is there. Here let's say a heavy substance is there. Okay, here there is lighter substance. Here there is lighter substance. Okay, students, obviously nam genetic material banbit, obviously ma'am genetic material will be heavier. So, we expect that the DNA or whatever the genetic material to be here, and we expect that whatever rest should be here. So, illi out check mada time ali. Okay, check mada time ali. See, look here, no radioactivity or no S35 presence in the cell. That means what? Protein will not enter the cell. Only genetic material will enter the cell. So, if protein does not enter the cell, the radioactivity will not be inside the cell. Alva. Next scenario dalli enaaytu, radioactivity detected in the supernatant. Supernatant is what? This one. So, remember two words you have to know here. This fluidy substance andre students idilva mud, that beach ka mud or any mud and water, if you mix nicely and you keep it there still, you keep it still, you will notice that on the top there will be water, on bottom there will be soil, correct? So, our soil will be called the pellet in centrifugation, I am telling. Soil will be called the pellet. What detected in the supernatant? Radioactivity is detected in the supernatant. No radioactivity detected in the supernatant. Radioactivity detected in the cell. Why? Because here genetic material got inside, that was radioactively labeled. So, we understood that okay, radioactivity got into the cell. So, obviously DNA is the genetic material. Any doubts here? Any doubts here? Huh? Come on. Doubts here? Come on, come on, come on, come on. Understood? Thumbs up. Only if you give me thumbs up, I'll move forward. Okay? Yes, ma'am. No doubts? Awesome, awesome. Clear to all of you? Perfect, perfect. Okay. Idu artha aitalla? Okay. So, students, ಈ ಒಂದು Hershey and Chase experiment ಇಂದ uni-equal proof andre, we ultimately understood that this is the this is the truth that DNA is the transforming principle. DNA is the one who transformed from the heat-killed S strain to the R strain. Alva, artha aita? So, adanna prove madake namage Hershey and Chase experiment bekagittu, because every McLeod and McCarty experiment bandbit solpa clarity kammi ittu. Every McLeod and McCarty experiment solpa clarity kammi ittu. So, adikke ನಾವು ಆ ಕ್ಲಾರಿಟಿ ಇಲ್ಲ ಒಂದು ಸಿಚುವೇಶನ್ ಇಂದ ಒಂದು ಫುಲ್ ಪ್ರಾಪರ್ ಕ್ಲಾರಿಟಿಯಲ್ಲಿ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡಿದ್ದೀವಿ. ಬಿಕಾಸ್ ರೇಡಿಯೋ ಆಕ್ಟಿವ್ ಲೇಬಲಿಂಗ್, ಸೆಂಟ್ರಿಫ್ಯೂಗೇಷನ್, ಈ ಥರ ಮಲ್ಟಿಪಲ್ ಪ್ರೊಸೆಸ್ ಇಂದ ನಾವು ಒಂದು ಅಲ್ಟಿಮೇಟ್ ಕನ್ಕ್ಲೂಷನ್ ಅಲ್ಲಿ ಬಂದಿದ್ದೀವಿ ದಟ್ DNA is the genetic material. Understood? All of you understood? So we proved it that DNA is the genetic material by using radioactive labeling, by using centrifugation. We had a clarity on what to be done. Perfect? Okay. Done. Okay? Yes. A molecule that can act as genetic material must fulfill the following criteria. Now let's say you are my student. Okay? Now let's say my students are the ones who come every day to my class. They are my strength, they are my, happiness, they make so much things for me. The only thing that I have given them is the proper content. Okay, I have connected them through online. I have not seen my students. But then a one connection build ಆಗಕ್ಕೆ ಒಂದು criteria ಇತ್ತು. ಏನಿತ್ತು ಆ criteria? ಅಂದರೆ ಅವ್ರು ಡೈಲಿ ನನ್ನ ಲೈವಲ್ಲಿ ಅಟೆಂಡ್ ಮಾಡಿದ್ದಾರೆ, ಐ ನೋ ದೇರ್ ನೇಮ್ಸ್, ನನಗೆ ಮೆಸೇಜ್ ಮಾಡಿ ಅವ್ರ ಪ್ರಾಬ್ಲಮ್ಸ್ ಹೇಳಿದ್ದಾರೆ, ಐ ಹ್ಯಾವ್ ಹೆಲ್ಪ್ ದೆಮ್ ಡ್ಯೂರಿಂಗ್ ದೇರ್ ಪ್ರಾಬ್ಲಮ್ಸ್. ಆ ಥರ ಒಂದು ಪ್ರೊಸೆಸ್ ಇತ್ತಲ್ವಾ? ಆ ಥರನೇ ಇಫ್ ಲೆಟ್ಸ್ ಸೇ DNA ಶುಡ್ ಬಿ ದ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್, ದೇ ಹ್ಯಾವ್ ಟು ಗೋ ಥ್ರೂ ಅ ಪ್ರೊಸೆಸ್, ದೇ ಹ್ಯಾವ್ ಟು ಗೋ ಥ್ರೂ ಅ ಲಿಟಲ್ like a procedure, or let's say they should go through this criteria to become the genetic material, because they are the genetic material for most of the organism. So ನಾವು ಇವಾಗ DNA ಮತ್ತು RNA ನ ಕಂಪೇರ್ ಮಾಡೋಣ. Okay, DNA ಮತ್ತೆ RNA compare ಮಾಡೋಣ. Okay, so if you have to be, let's say I'm telling DNA, if you have to be the genetic material, the first thing that you need to do is you should be able to do replication. Okay, you should be able to do replication. You should be able to do what, students? You should be able to do replication. Correct? Replication ಮಾಡಬೇಕಂತ. So DNA was like, okay, so we have DNA, we have RNA. So whoever gets the maximum tick, okay, they are going to win this race. So DNA was like, okay, I'll do replication. Even RNA was like, I'll do replication. Okay, so replication andre making their own replicas. Okay, ಇಲ್ಲಿ ಬರೀತೀನಿ. Students, my notes, no, everything will be there. So ನನ್ನ notes blindly follow ಮಾಡಿ ನಿಮಗೆ ನೋಡ್ಬೋದು. For Karnataka students ಈ ಥರ ಒಂದು detail lecture ನಿಮಗೆ ತುಂಬ ಕಮ್ಮಿ ನೋಡೋದು. Most of them NCERT snips use ಮಾಡ್ಬಿಟ್ಟು underline ಮಾಡ್ತಾರೆ. I'm not telling that teachers are bad or anything, but it's their situation. But here we have the ability. Vedantu wants to give Karnataka students quality education. ಅದಕ್ಕೆ ಈ ಚಾನಲ್ ಸ್ಟಾರ್ಟ್ ಮಾಡಿರೋದು. So I can 100% say that ನಿಮಗೆ ಈ ಥರ ಡೀಟೇಲ್ ಆಗಿ ಈ ಥರ ರಿಟರ್ನ್ ನೋಟ್ಸ್, okay, like this hand written notes you will not be able to find. That is why I'm telling you, follow the channel. If your goal is to write NEET, you want to go for any other coaching, you go for any other coaching, no problem. But ಏನಾದರೂ ಒಂದು ಡೌಟ್ ಇದ್ರೆ, ಏನಾದರೂ ಒಂದು ಟಾಪಿಕಲ್ ಕ್ಲಾರಿಟಿ ಕಮ್ಮಿ ಇದ್ರೆ, come back here. Okay, come back here and watch the videos, take the notes and study happily. Okay, study happily. So here you see, replication means making their own replica. Okay, making their own replica. Okay, so they were like, both of them were like, it's okay, I will also do, you also do. Okay, now here second thing that these people told was mutation criteria idu. Okay, replication ಮಾಡಬೇಕು, mutation ಮಾಡಬೇಕಂತ criteria ಕೊಟ್ಟಿದ್ದಾರೆ. So DNA told, okay, I will do slow mutation, but RNA is like, no, I will do fast mutation. Why he'll do fast mutation, students? Because it is very reactive, alva. So RNA bandbit, it's highly reactive. RNA is highly reactive. DNA is not reactive. Do anyone wants to sleep, like let's say I want to sleep as a human, tomorrow I want to wake up as a giraffe? No ma'am, I don't want that fast mutation to happen. So mutation ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ ಸ್ಲೋ ಆಗಿ ನಡಿಬೇಕು. ನಾವು ನೋಡಿದ್ರೆ ಜನರೇಷನ್ ಟು ಜನರೇಷನ್ನು ಡಿಫ್ರೆನ್ಸ್ ಇರಲ್ಲ, right? All the humans have the same characteristics. Millions of years later maybe ಒಂದು ಮ್ಯೂಟೇಷನ್ ಬರುತ್ತೆ. ಆ ಥರ like mutation should be very very slow. We do not want to sleep like a human, wake up like a giraffe. I'm just giving example, okay? So ಆ ಥರ we don't want. So slow mutation, fast mutation. So here DNA is going to get 1 point. Okay, DNA is going to get 1 point. DNA approved, RNA not approved. Third thing that these people told was, see, chemically and structurally stable also matters a lot. Okay, chemically and structurally stable. Because students, very simple. We don't want our DNA material, right? Let's say temperature change, environmental, like let's say normally students, ನಮಗೆ ಇವಾಗ ಬ್ಯಾಂಗ್ಳೂರಲ್ಲಿ ಒಂದಿನ, ಓಕೆ, ಒನ್ ಡೇ, ಇವತ್ತೊಂದಿನ 3 ಸೀಸನ್ಸ್ ನೋಡುತ್ತೆ. ಅಂದರೆ ಮಾರ್ನಿಂಗ್ ಸಮ್ಮರ್ ಇರುತ್ತೆ, ಆಫ್ಟರ್ನೂನ್ ಇಟ್ ಬಿ ರೈನಿ, ಈವ್ನಿಂಗ್ ಇಟ್ ವಿಲ್ ಬಿ ವಿಂಟರ್. ನಾವು ಈ ಥರ ಎನ್ವಿರಾನ್ಮೆಂಟಲ್ ಚೇಂಜಸ್ ಕೀಪ್ಸ್ ಹ್ಯಾಪನಿಂಗ್. Now let's say my genetic material is like, oh, today is winter, I want to change. Today morning is summer, I want to change. It cannot be affected by the environmental changes. Let's say, okay ma'am, DNA does not get affected. But let's say due to some accident, a body burn happened or something happened. We do not want a genetic material to be reactive. We do not want a genetic material to, get destroyed completely. So genetic material should be someone who is chemically and structurally very stable. Very sorted fellow he should be. So when you look at this, ನಿಮಗೆ already answer ಗೊತ್ತು. All of you tell me who is going to get the tick. All of you tell me who is going to get the tick. ಬೇಗ chat box ಅಲ್ಲಿ, come on. Who is going to get the tick? Will DNA get or will RNA get? Who is going to be the winner of the competition? Who is going to be the winner of the competition? Ma'am, I will be the winner, ma'am. Both are not going to be winners. I will be the winner. Don't say that. Don't say that. Okay? Who is going to be the winner? I will be the winner. Yes, very good. Excellent. So, DNA is the one who is going to get the tick. RNA is not going to get the tick. Why? Because students, here we already know RNA is not chemically or structurally stable. Why? Because of 2 reasons. Okay? Oh, yes. This is a NEET question, assertion reason question. What are the reasons like RNA is unstable anta? So first reason is the presence of O group. Okay, presence of oxygen. Also one of the reason yak RNA unstable ಆಗಿರೋದು. Yes, RNA is unstable, ma'am. Good job. So DNA gets 1 tick, RNA gets 1 cross tick. Okay. Last one, students, they should be able to express themselves in Mendelian characters. Okay, Mendelian characters. They should be able to express themselves in Mendelian characters. Both of them will do it. So Mendelian characters, ನಿಮಗೆ already principles of inheritance ಅಲ್ಲಿ ಅರ್ಥ ಆಗಿರ್ಬೋದು. So they should be able to express themselves. Yes, both of them will express themselves. So if you look here, RNA is 2 points down, DNA is 4 points up. So obviously who is going to be the genetic material? DNA is going to be the genetic material for majority of the organisms. Then you will be like, ma'am, what about RNA, ma'am? RNA doesn't have a life or what? Students, RNA has a life, okay? See, we are not going to ignore anyone, okay? Ratnam Institute of Technology, Bangalore. Wolf Gaming, I have not heard much about the institute here. Check ಮಾಡಬೇಕು. ಬಿಕಾಸ್ ಎಲ್ಲ ಕಾಲೇಜ್ ಬಗ್ಗೆನೂ ಯಾರಿಗೂ ಗೊತ್ತಿರಲ್ಲ ಸ್ಟೂಡೆಂಟ್ಸ್. ಟೀಚರ್ಸ್ ಆದರೆ ಕೂಡ ಯಾರಿಗೂ ಗೊತ್ತಿರಲ್ಲ. ಕೌನ್ಸಿಲಿಂಗ್ ಅವ್ರಿಗೆ ಗೊತ್ತಿರ್ಬೋದು, ಓಕೆ, ಬಟ್ ನನಗೆ ಗೊತ್ತಿಲ್ಲ ಅಷ್ಟು ಕ್ಲಿಯರ್ ಆಗಿ. ಐ ನೋ ಅಬೌಟ್ BMS, PES, ದಯಾನಂದ ಸಾಗರ್, ದಿಸ್ MSRIT, RV ಕಾಲೇಜ್, ಆ ಥರ ಆಲ್ಮೋಸ್ಟ್ ಸಮ್ ಕಾಲೇಜಸ್, ವಿಶ್ವೇಶ್ವರಯ್ಯ. ಸೊ ಲೈಕ್ ದ್ಯಾಟ್ ಫ್ಯೂ ಕಾಲೇಜಸ್ ಐ ನೋ. ಓಕೆ, ಇದನ್ನು ಚೆಕ್ ಮಾಡಿಬಿಟ್ಟು ಹೇಳ್ತೀನಿ. ಓಕೆ, ಎಸ್. ಸೊ ಎಸ್, ಐ ಹೋಪ್ ಆಲ್ ಆಫ್ ಯು ಆರ್ ಮಾರ್ಕಿಂಗ್ ಯುವರ್ ಅಟೆಂಡೆನ್ಸ್. ಕಮ್ ಆನ್, ಸ್ಮ್ಯಾಶ್ ದ ಲೈಕ್ ಬಟನ್. ಓಕೆ ಓಕೆ, ಇಲ್ಲಿ ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್. ಸೊ ನೀಟ್ ಸ್ಟೂಡೆಂಟ್ಸಿಗೆ ಲೆಟ್ಸ್ ಸೇ ನಿಮಗೆ ನೋಟ್ಸ್ ಬೇಕಂದರೆ ನನ್ನ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲು ಫಾಲೋ ಮಾಡ್ಬೋದು. ನನ್ನ ಹೆಸರು ಟೈಪ್ ಮಾಡಿ, ಬಯಾಲಜಿ ಅಂತ ಟೈಪ್ ಮಾಡಿ, ಒಫಿಷಿಯಲ್ ಅಂತ ಟೈಪ್ ಮಾಡಿದ್ರೆ ನನ್ನ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್ ಓಪನ್ ಆಗುತ್ತೆ. ಅಲ್ಲಿ ನಿಮಗೆ ಲಾಸ್ಟ್ ಇಯರ್ ಎಂತಿರುವ ಕ್ವೆಶನ್ಸು ಮತ್ತು ನೋಟ್ಸು ಎಲ್ಲ ಇರುತ್ತೆ ಆ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್. ಓಕೆ, ಸೊ ಅದನ್ನು ಜಾಯಿನ್ ಮಾಡಿ, ಸೊ ದಟ್ ಯು ಕ್ಯಾನ್ ಡೌನ್ಲೋಡ್ ನೋಟ್ಸ್, ಯು ಕ್ಯಾನ್ ಡೌನ್ಲೋಡ್ ಆಲ್ ದ ಇನ್ಫಾರ್ಮೇಷನ್ಸ್. ಕ್ಲಿಯರ್? ಸೊ ನನ್ನ ಹೆಸರು ಟೈಪ್ ಮಾಡಿದ್ರೆ ನಿಮಗೆ ಆಟೋಮ್ಯಾಟಿಕ್ಲಿ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್ ಯು ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ಫೈಂಡ್ ಇಟ್. ಓಕೆ, ಇಲ್ಲಿ ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್, a molecule that can act as a genetic material. So I told you, now it is confirmed that DNA is the genetic material. DNA is the genetic material. DNA is the genetic material. Now here what you have to do, what about RNA? So RNA is a genetic material. RNA is a genetic material. RNA is a genetic material for which organisms? For few viruses. Okay, few viruses like your HIV, TMV, okay, coronavirus, atara virus igella, this is the catalyst. It also acts as messenger. It also acts as messenger. So, itara solpa kelsa the RNA is going to do. It is not going to be like, okay, I am just a genetic material for few substance, so I am going to be sad, I am going to cry forever, atara eno ego complex illa. So, it is like, no, I will help in other processes like acting as an adapter molecule, acting as a catalyst, acting as a messenger. I will do all those jobs. Okay. I'll do all those jobs. This is clear? Any doubts here? Okay, nodi. The genetic material should be stable enough. See, stable enough to change, to not change with the different stages. Okay, different stages of life cycle. Life cycle like childhood alone DNA, adult alone DNA, teenage alone DNA, atara change madakagalla. Okay, amele age or with change in physiology of the organism. Okay, physiology of the organism. Now, stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself, that the heat which killed the bacteria at least did not destroy. See, at least did not destroy some properties of the genetic material, right? That is the main reason, students, why our naked DNA pick up madi, okay, our strain became virulent. Understood? ಲ್ಯಾಗ್ ಆಗ್ತಾ ಇದೆಯಾ? ಲ್ಯಾಗ್ ಆಗ್ತಾ ಇದೆಯಾ? ಒಂದು ನಿಮಿಷ ಚೆಕ್ ಮಾಡ್ತೀನಿ. ಪ್ರಾಬ್ಲಮ್ ಇಲ್ಲಲ್ಲ? ಕ್ಲಿಯರ್ ಆಗಿದೆ. ಅನಿತಾ, ಜಸ್ಟ್ ರೀಜಾಯಿನ್ and see. Just leave the channel, just rejoin and see. ಒಂದು ನಿಮಿಷ ಚೆಕ್ ಮಾಡ್ತೀನಿ ಇಲ್ಲೂ. Done. Problem ಏನು ಇಲ್ಲ. Okay, correct ಆಗಿದೆ. Maybe it was just for a minute. Okay, just for a minute. Okay, done. Work ಆಗ್ತಾ ಇದೆ ಅಲ್ವಾ? Awesome. Okay, yes. So here, this is now can easily be explained in the light of the DNA that two strands being complementary if separated by heating come together. Students, you understood? Now we know, okay, one second, complementary, complementary if separated, it's going to come together. So andre, let's say illon DNA strand ide, illon DNA strand ide alva. Let's say nan heat kotti idanna separate madidini. After some time also ಅವ್ರಿಗೊಂದು tendency ಇರುತ್ತಲ್ವಾ to come back together. Obviously students, they are complementary to each other, they will come back together. So because they come back together, there is chances that their properties, that nature of getting inherited, our nature irutte. Okay, now when appropriate conditions are provided. Further, the OH minus group, okay, present at every nucleotide in the RNA is a reactive group. This I already told you yesterday. It's a reactive group and makes the RNA liable and easily degradable. RNA is also known as a catalytic. Okay, RNA is also known to be catalytic, hence reactive. So RNA also has a catalyst nature, that is they will increase the rate of the reaction but does not participate in the reaction, right? So they have a catalytic nature, because of this they will be highly reactive. Therefore DNA chemically is less reactive and structurally more stable, because avrige reactivity illa. because our oxygen group illala, so because of that they are very stable, they are like very calm. Okay, now when compared to RNA, therefore among the two nucleic acid the DNA is a better genetic material. Among the two nucleic acid DNA is a better genetic material. We already proved it alva. ಇಲ್ಲಿ ನೋಡಿ ಈ ನೋಟ್ಸ್ ಇಂದ ನಿಮಗೆ ಕ್ಲಿಯರ್ ಆಗಿ ಅರ್ಥ ಆಗುತ್ತೆ ವಾಟ್ ಇಸ್ DNA, ವಾಟ್ ಇಸ್ RNA ಅಂತ. ಓಕೆ, RNA ವರ್ಲ್ಡ್ ಇಸ್ ಅಂಡರ್ಸ್ಟೂಡ್. ಓಕೆ, RNA ವರ್ಲ್ಡ್ ಇಸ್ ಅಂಡರ್ಸ್ಟೂಡ್. ರೆಪ್ಲಿಕೇಶನ್ ಈ ಕ್ಲಾಸಲ್ಲಿ ಮಾಡಬೇಕಾ, ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸಲ್ಲಿ ಮಾಡಬೇಕಾ? ಹಾಂ, ರೆಪ್ಲಿಕೇಶನ್ ಈ ಕ್ಲಾಸಲ್ಲಿ ಮಾಡಬೇಕಾ, ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸಲ್ಲಿ ಮಾಡಬೇಕಾ? ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸಲ್ಲಿ ಮಾಡೋದಾ ರೆಪ್ಲಿಕೇಶನ್? ಬಿಕಾಸ್ ರೆಪ್ಲಿಕೇಶನ್ ಸ್ಟಾರ್ಟ್ ಮಾಡಿದ್ರೆ ಮಿನಿಮಮ್ ನನಗೆ ಒಂದು 45 ಮಿನಿಟ್ಸ್ ಬೇಕಾಗುತ್ತೆ ಅದನ್ನು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡೋಕ್ಕೆ. ಅದಾದಮೇಲೆ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಮಾಡ್ತೀನಿ. ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ಸ್ವಲ್ಪ ಲಾಂಗ್ ಕ್ಲಾಸ್ ಮಾಡೋಣ, ಓಕೆನಾ? ಪ್ರಾಪರ್ 2.5 ಅವರ್ಸ್ ಕ್ಲಾಸ್ ಮಾಡಿದ್ರೆ ಇನ್ನೂ ಸ್ವಲ್ಪ ಕ್ಲಾರಿಟಿ ಇರುತ್ತೆ ನಿಮಗೆ. ಓಕೆ, ಬಿಕಾಸ್ ಇವಾಗ ಸ್ಮಾಲ್ ಕ್ಲಾಸಸ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ಮ್ಯಾಮ್. ಓಕೆ, ಡನ್. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್. ಸ್ಟೂಡೆಂಟ್ಸ್, ಇನ್ನೊಂದು ಇಂಪಾರ್ಟೆಂಟ್ ಥಿಂಗ್ ಇವಾಗ ನೋಡಿ, ಇಲ್ಲಿ those students who are participating for KCD counseling, ಇಲ್ಲಿ ವಿಡಿಯೋ ಸೆಷನ್. ಓಕೆ, ಗೋ ಟು ವಿಡಿಯೋಸ್. ಇಲ್ಲಿ ಇವಾಗ ರೀಸೆಂಟ್ ಆಗಿ ಒಂದು ವಿಡಿಯೋ ಮಾಡಿದ್ದೀನಿ ಅಬೌಟ್ ದ ಕಾಷನ್ ಡೆಪಾಸಿಟ್, BAMS, ಚಾಯ್ಸ್ 2, ಚಾಯ್ಸ್ 3 ಬಗ್ಗೆ. ಸೊ ಎಲ್ಲ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೀನಿ ಇಲ್ಲಿ. ಜಸ್ಟ್ ಚೆಕ್ ಔಟ್ ದಿಸ್ ವಿಡಿಯೋ. ಇನ್ ದಿಸ್ ವಿಡಿಯೋ ಐ ಹ್ಯಾವ್ ಟೋಲ್ಡ್ ಆಲ್ ದ ಇನ್ಫಾರ್ಮೇಷನ್. ಸೊ ಇಲ್ಲೆಲ್ಲ see some informations I have used to explain it. Clear? So just go watch this video. If your friends or anyone is there who is last year KCT ಬರ್ದಿದ್ದಾರೆ, you can show this. Okay, yes, KCT ಇದೆ, KCT. okay, you want a break? No, sorry. Okay, students, biology KCT ಗೆ ಇದೆ ನೀಟ್ ಕಂಟೆಂಟ್ ಯೂಸ್ ಮಾಡ್ಬೋದು. ಓಕೆ, ಯು ಡೋಂಟ್ ಹ್ಯಾವ್ ಟು ವಾಚ್ ಸಪ್ರೇಟ್ ವಿಡಿಯೋಸ್. ಈ ವಿಡಿಯೋ ಸೆಷನಲ್ಲಿ ನಿಮಗೆಲ್ಲ ಇರುತ್ತೆ. ನೀಟ್ ಸ್ಟ್ರಾಟಜಿ ವಿಡಿಯೋನು ಬೇಗ ಮಾಡ್ತೀನಿ ನಿಮಗೆ ಏನಾದರೂ ಹೆಲ್ಪ್ ಬೇಕಂದರೆ. But other than that, everything is basically KCT videos. So if you want any help from here, you can watch this video. Subscribe to the channel, and also if you have any brothers or sisters, just search Vedantu Karnataka. Okay, 10th. Okay, 10th barala. Okay, 1 second. Vedantu Karnataka SLC you type. Okay, state board SLC if you type, you will get a channel here. Okay, see, you can see here Vedantu. Yes, here Vedantu SLC Karnataka. This channel ಇವಾಗ ಲೈವ್ ನಡೀತಾ ಇದೆ ಅನಿಸ್ತಾ ಇದ್ದೀನಿ. ಓಕೆ, yes, live is happening. So here you can see state board. Okay, state board classes are happening. ICC classes are also happening. So if you have any siblings or anyone, you can subscribe to this channel. Okay, clear students? Understood? Okay, awesome. So thank you so much. See you all in next class. I hope all of you like the class. Please do like the video and also subscribe to the channel, and let me know in the comments how the class was. Okay, otherwise how will I know? Okay, so yes students, so see you all. Bye bye, take care. ಈ ನೋಟ್ಸ್ ನಿಮಗೆ 15 ಮಿನಿಟ್ಸಲ್ಲಿ ಅಪ್ಲೋಡ್ ಮಾಡ್ತೀನಿ. ಓಕೆ, ಮ್ಯಾಥ್ಸ್ ನೋಟ್ಸ್, KCT ಮ್ಯಾಥ್ಸ್ ನೋಟ್ಸು ಮತ್ತು ಫಿಸಿಕ್ಸ್ ನೋಟ್ಸು ಅಪ್ಲೋಡ್ ಮಾಡಿದ ಟೀಚರ್ಸ್ಗೆ ಹೇಳ್ತೀನಿ. And we will also possibly share all these notes with you on a sheet, okay, and a proper Excel sheet where you can get access to all the notes. Okay, don't worry about notes and anything. ನೀವು ಏನಾದರೂ ಕಮೆಂಟಲ್ಲಿ ತಿಳಿಸಿದ್ರೆ immediately we will check and we'll solve the problems then and there. Okay, so thank you so much.